good morning, good evening, good afternoon, wherever you are, and many thanks for joining this exciting symposium today. So I am a diabetes specialist in practice and also a clinical academic, and I work in the north of England in Sheffield. And it's my honor to chair the Grand Challenge on behalf of the Steve Morgan Foundation and the two coordinating charities, Diabetes UK and the JDRF. Steve Morgan is a builder who's been very successful working in the northwest of England, and he runs a foundation which does great charitable work, generosity in terms of the funding for a number of different good causes. But this is the purpose of the Grand Challenge, is to hasten the cure and improve the treatment of type 1 diabetes. And we are very grateful to Steve and his wife Sally for their generosity in providing a huge amount of money in the UK to do this. He has a son who has type 1 diabetes, which may be at least one of the motivations, but it's an opportunity for us, as they both have said, to accelerate the progress and harness the brilliance of UK science in this direction. He and Sally chose three themes in which to focus on, and one is cell therapy in type 1 diabetes, one is the causes of type 1, and the third is novel insulins, and seeing if we can increase the speed at which we develop insulins, which could make a real difference. And the foundation is offering 15 million. And today's meeting is the start of a two-stage process, if I can call it that. And what we hope to do over the next few hours is conduct a state-of-the-art symposium, listening to experts from all over the world. And this will be followed by an invitation for consortia to get together, having listened to what's been said today. And of course, many of them will have been working on this project for many years. And we will invite people to pitch, if you like, the ideas in a meeting to happen a little later on in the year. And Rachel Connor will talk about that towards the end. So with that, I want to invite Jonathan Rosen, who I hope has joined us from the US, to say a few words on behalf of the JDRF. Oh, good to see you, Jonathan. Thanks for joining.
Hi, Simon. I am indeed here. Good to see you, and thank you for the introduction. My name, for those of you who don't know me yet, is Jonathan Rosen, and I'm a scientist at JDRF International. I work in our Improving Lives program, which is about developing drugs and devices and even behavioral health interventions to improve outcomes for people with type 1 diabetes. On behalf of JDRF's global community, I'd like to thank Steve and Sally Morgan and their fellow trustees at the Steve Morgan Foundation for their investment in type 1 diabetes research. I must also acknowledge the superb group of international speakers that JDRF UK has gathered for today's symposium. Thank you all so much for taking the time. I must also thank the Scientific Advisory Panel for their support with the Novel Insulins Grand Challenge. Thank you to Dr. Simon Heller, Reinhard Becker, John Buse, Tim Heise, David Kelley, and Diane McCarthy. And thank you so much to my colleagues at JDRF UK for leading this project. I'm sure that today will enable interesting and thought-provoking ideas to be shared, and I hope it'll prove a catalyst for proposals that will take our research into novel insulins further forward more quickly in order to benefit those living with type 1 as swiftly as possible. Development of insulins is a major priority for JDRF. I think we all know that most people with type 1, especially young people, aren't hitting their A1C and other recommended glucose control targets. And as a result, people are still at risk of long-term complications and acute complications like hypoglycemia. And today is a chance to really get the ball moving to do something about this. So thank you, everybody.
Thank you, Jonathan. really appreciate it. Now, as I said at the moment, although I'm a clinical academic and have the honor to chair the Grand Challenge, it will have occurred to you that I am no expert in this. And indeed, what we have in all three themes are people who've agreed to vice chair, and they're the real experts, particularly in terms of the groups of experts they brought with them for these three areas. And I want to just, before I introduce the first speaker, thank Tim Heise. He's a friend and colleague who I've known for many years, and his generosity, and indeed those of his colleagues, some of whom will be chairing some of the talks this afternoon, is both humbling and I think on behalf of the whole Grand Challenge, it's important for me to thank them for giving up their time and expertise to aid us in this endeavor. So I want to start now by introducing Dr. Peter Kurtzhals. I've known Peter for many years. I regard him as a friend and again as a colleague who I've worked with. He is Chief Scientific Advisor for Novo Nordisk R&D, and he's been a coordinator for their insulin development project over the years, including development of insulin aspart, detamir, and insulin degludec. I can't think of any better person to introduce this symposium. It's with great pleasure I asked Peter to give his presentation today. I should say he's going to talk for 20 minutes, and there'll be an opportunity for you to ask questions, which I'd like you to post in the chat. And I'll do my best to conduct a 10-minute question and answer at the end of this presentation, as will the other chairs. Peter, over to you.
Thank you very much for giving me an opportunity to present here today and to be part of this very interesting meeting. I understand that a lot is going on with this new grant opportunity within the support to people with type 1 diabetes long term. Personally, I've been involved with insulin research and diabetes research in some shape or form from an industrial point of view for 33 years. And I've had an interest, of course, in insulin in particular, since I represent also the company that is producing half of the world's insulin at this time. So we've been celebrating our 100 years anniversary in 2023 as a company here in February, and the challenge remains in further improving insulin therapy. It has been a kind of pearls on a string of examples of how industry has been building new product developments on top of discoveries made in academia when you look at the insulin field. All the way from the beginning in 1923 with the discovery of insulin, in 1921 with the discovery of insulin, followed by the industrialization of insulin production shortly thereafter. We've seen stepwise improvements of insulin therapy, and often guided and made possible, enabled, you can say, by discoveries in academia. And insulin itself has been kind of the cornerstone of a little less than a handful of Nobel Prizes. That also tells you about the importance of this molecule, not only for therapy, but also as a model example of a protein substance used in pharmacology. And it's still true that we need to work together to get to improvement of therapy in the future. Of course, it's given that it's a collaboration when it comes to clinical research, but even for basic research, we also build on each other's learnings and publications and interactions like the one that we have here today as well. Then I think it's important for everyone listening in here today that to understand that the road to commercialization and bringing a product to the patients is long. Still for a new insulin molecule, we say here that average time of development will be 10 to 12 years from embarking on a research idea until a successful project has gone all the way through the various stepwise processes in the R&D process and gets all the way out to the patient. So it's not something that happens quickly. I showed a picture of the Danish Nobel Prize winner, August Chou, who was one of the founding fathers of the Novo Nordisk company. And when he returned from Toronto in 1922 with the right to produce insulin in Scandinavia, it was less than 3 months from that date until they treated the first Danish patients with insulin produced in Denmark. So that was a time window of 3 months from the very first crude experiments and to treatment of the first patient. And we're talking about years and years as we speak, and the regulatory requirements are still increasing for new pharmacological modalities. So it's important to be aware of that timeline as well. And if we look a little bit closer into an example, the last major submission of a new insulin product that we made from Novo Nordisk was insulin degludec, Tresiba. Goes back more than 10 years now. But if you stack the paper on top of each other that was part of that submission, it's 1.5 kilometers of paper with the numbers that you see there. It's a huge undertaking to make a regulatory submission ready to take place. So it doesn't come easily. We need to work together, and those who are working with the regulatory submissions also need to be involved, of course, to make it right, that we get the right documentation in place, both for efficacy and for safety and for production and so forth. But I wanted to start with that until from the beginning here, until we now jump more into the science of insulins. So when we design insulin analogs, as we've been embarked with or involved with for more than 30 years, I joined Novo Nordisk when it was just in its upcoming. The first publication of a rapid-acting analog was in Nature in 1988 from Novo Nordisk. It's all about improving pharmacokinetic properties to make it possible to mimic as closely as possible the natural patterns of insulin release as we have them in non-diabetic individuals. Then in the future, there may be other benefits that we want to build into novel analogs to improve therapy. But from the beginning, it was about improving kinetics. And in doing that, we need to preserve efficacy and safety of the molecule. And we also need to take care of pharmaceutical formulation. And that means the pharmaceutical properties such as stability, solubility, possibility for combining between rapid and long-acting analogs or between insulins and other types of molecules for pharmacological action needs to be considered as well. And so does the possibility to express or produce the candidate insulin analog can make a whole lot of difference, not only to pharmacology, but also to production cost, of course, but also to environmental impact, such as energy utilization, water utilization, generation of waste, which are also important parameters in choosing a development candidate these days, apart from the pharmacological optimization. So a lot of things need to come in place and together for making a new analog a good candidate for further clinical development and bringing it to the patients. Then we've taken a lot of inspiration in designing analogs from what goes on in nature. And you have insulin there with its A chain and its B chain, with 21 amino acids in the A chain, 30 amino acids in the B chain, and linked by 2 disulfide bridges. And as you can see, the alphabet, so to speak, of an insulin depends somewhat on the species that we are looking at, with minor differences to, for example, to pig insulin only differing from human insulin in the B30 position, and other animal insulins showing greater variability compared to human. And that tells us something about areas in the molecule that may be suitable for modification in the design of new insulins, and also to areas of the insulin molecule that may be less suitable because they probably play critical roles for structure, stability, or action of the hormone. So that the areas that are preserved in nature, indicated here with the gray columns, is probably areas that should be left untouched in designing new analogs, whereas the areas where you see the flexibility in nature are more flexible probably and therefore also areas that are amenable to substitution of amino acids to obtain certain benefits in analog generation. And that has been taken into consideration also, of course, in designing novel insulins. Then there are two fundamental properties that we need to take care of. and consider in designing analogs at least two. One is the self-association of the insulin molecule, which is an inherent property of the natural hormone, and it's also occurring in nature. Insulin is stored in the pancreatic beta cells in the hexameric form, with the zinc coordinating the 6 insulin molecules together. And you can see the amino acid sequences color-coded here involved in the dimer formation or in the hexamer formation of the insulin molecule. So the formation of hexamers is important for structural stability, but is also critical for time action profile of insulin following subcutaneous injection, because the more stable, larger molecular size of the insulin hexamer makes it a more slowly absorbed molecule than the monomer, which is a biologically active species. So we have to consider the balance between the monomer and the hexamer in designing an analog preparation with the right desired kinetic profile. So what comes as a benefit for stability can become a limitation, for example, when it gets to designing ultra-rapid acting insulin analogs, namely the hexamer formation. Then another fundamental parameter that needs to be taken into consideration is receptor binding and recognition by the insulin receptor. Of course, you need to retain receptor binding and recognition and activation of the receptor in new analogs for them to be pharmacologically useful. However, receptor binding and receptor affinity also has to do with the kinetics, because insulin is cleared via its insulin receptor, so that a reduction of relative receptor binding affinity leads to a longer circulation half-time, and a faster elimination will occur with insulins that have a greater affinity for the insulin receptor. So the receptor binding, understanding of receptor binding properties of a new analog are critically important, both for overall pharmacological action, but also for time action profile. And that's quite important to understand that as well. Now we are in the privileged situation that we have an understanding of the structural interaction between the insulin monomer and its receptor, as indicated in this slide here. So self-association, receptor binding are critical parameters that need to be taken into account when we design novel analogs. What is it then that is cooking in the labs of Novo Nordisk as we speak? Well, here is a handful of examples of what we are thinking may be of benefit for people with type 1 and or type 2 diabetes in the years to come. One is the holy grail of an insulin with a built-in glucose sensitivity, glucose-sensitive insulin that would target reduction of hypoglycemia risk. One is a tablet-based product that could be an ultimately convenient product, at least for people with type 2 diabetes, but maybe even with type 1 diabetes subjects as a target. Then we have an improved pump insulin in our pipeline to hopefully be useful in closed loop systems as we get closer to that. Once weekly insulin to reduce the number of basal insulin injections by more than 300 injections per year, from 365 to 52. And then we have this last opportunity of tissue or pathway targeted action, where we're moving away from insulins that are just optimized for kinetic properties, but are also optimized for some kind of biological upside. And I'll come back a little bit to that as well. But that's a kind of a box, I can say a grayish box at the moment. And we are working with some ideas in that realm as well, for what we call bio-age insulins with a biological age other than just a kinetic benefit. But let me give you a few more glimpses into these various project opportunities. First, we're working with the faster acting insulins. We know aspart has been around for almost as long as Lispro. In the field, we've got faster versions of both aspart and Lispro also for the patients. But we still need to have that insulin that is rock stable in the pump, and it's also even faster in its onset and offset of action to be useful in closing the loop. And we've been working with that quite dedicated for some years, and we now have an insulin that we call insulin 119 in early clinical trials, and we believe that insulin will fulfill some of those properties and hopefully be an insulin that's providing a more time in range because of its faster action than the current versions of fast-acting insulin preparations built on the known substances with Aspart and Dispro. So this is a novel insulin analog, and we'll have to go through the full regulatory package to get to an approval. It's not just a formulation approach, it's a novel insulin analog with all that it takes for characterization of both pharmacology and toxicology. We're not disclosing, unfortunately, any details of this at this time. Another project that is far more progressed is the project on insulin, once weekly insulin in the basal segment, and we've gradually or stepwise moved towards greater and greater duration of action, as you can see in this slide here, coming from NPH insulin, which is still pretty good actually, 75 years after its discovery in 1946 by Hagedorn, as you see in the picture there. We then came to the first generation of analogs with Delamere and Lantus, and to Degludec with the second generation, so to speak, basal insulin analogs, and now with moving all the way to once weekly, which many of us had thought was impossible if you just go a few years back. But now it seems to be an opportunity, and we have filed for regulatory approval in several countries already now. The candidate that we have developed at Novo Nordisk is called insulin icodec. You see it in the right part of the picture there. It's just carrying a fatty acid tail, but otherwise retaining the majority of the primary structure of human insulin. And then we have the Lilly candidate, as you see to the left in this slide, which is a fusion protein where two insulin molecules have been fused onto an Fc domain. And it's also showing a very long duration of action, actually a half-life of a couple of weeks, whereas insulin icodec has a half-life of 1 week in humans. Icodec was done based on, or engineered based on the knowledge of self-association and receptor binding parameters and albumin binding parameters, as well as we've learned over the years. And as you see in the left panel of part of this slide here, the way to protract icodec action has been to combine an increasing strength of albumin binding facilitated by the C20 diacid attached to position B29 of the molecule, but to combine that with a reduction of insulin receptor affinity, which sounds counterintuitive, but this is exploiting this property of insulin receptor binding that it also leads to elimination. And therefore, when you reduce receptor affinity, you get to a lower rate of elimination of insulin from the circulation. And so when you combine albumin binding and low receptor affinity, you get to an ultra-long duration of action due to a circulation half-life increase. So this is an insulin that's mainly protracted in the circulation and not at the site of injection. And in phase 3, the icodec has shown promising results, and we're now in the regulatory discussion phase with this insulin. But that leads me to talking about insulin 338, which is a closely related analog of icodec and basically done with the same principles. And I would say icodec, or the once weekly opportunity, actually grew out of our interest in developing an oral insulin. And because for an oral insulin, as you can see in the right panel here, you would have to have a long circulation half-life in order to make basal insulin action. So when you eat a tablet or take a tablet or a capsule, absorption will be quite fast. So to provide a basal insulin action, you have to have a long circulation half-life and kind of push the protraction into the into the bloodstream and not at the site of intake. Whereas in subcutaneous preparation, it's typical that we've had the long retention time at the subcutaneous tissue and then a fast kinetics once in the bloodstream, fast recognition by the receptor and elimination. So this the enabling parameter in icodec was also the enabling parameter for the oral insulin projects, namely to produce that mechanism of protraction that would lead to protraction in the bloodstream. And that's what we got with insulin 338, due to that combination of strong albumin binding and slow clearance by insulin receptors. We got to a half-life of 70 hours with 338. icodec is 160 hours, so good for once weekly. But we thought insulin 338 would be a strong candidate for once daily oral dosing. And when you give an oral dosing of a compound with a long half-life, you get to a very flat profile in steady state. And together with Tim Heise's group, we did this quite, I think, exceptional study in showing that a daily tablet of insulin 338 was as effective in lowering blood glucose as insulin glargine given once daily as subcutaneous injections. Unfortunately, the dose that we needed for insulin 338, despite being formulated with an absorption enhancer, was still around 50-fold greater than that of insulin glargine, subcutaneous glargine, and that was deemed to be non-financially viable by the organization here. And therefore, we have been looking for opportunities to further increase the bioavailability of insulin and insulin like 338, but still having the basal action due to that formulation that engineering principle that I was just taking you through for 338. So we've been working with Bob Langer's group, and I know Bob is speaking later today, in designing an oral device that would lead to a gastrointestinal injection of an insulin millimeter scale pellet into the gastric wall, and therefore increasing bioavailability quite significantly from oral delivery. But moving quickly to the next couple of ideas, one was that of a bioedge. So insulin given subcutaneously is not really physiological in its distribution in over-insulinizing the muscle and giving too less activity at the liver. And we've designed insulin analogs that would combine a very high insulin receptor affinity with a strong albumin binding. And when you combine these two parameters, you get to a preferential uptake in the liver of an insulin. And that was exactly what we did in designing an insulin called 406, where, as you can see in the right panel here, you get to around 40% of insulin action in the liver compared to only 25% for insulin aspart. So in that regard, we almost restore a normal distribution of insulin action distributed between the liver and the periphery, as we know it from portal delivery of insulin in a normal, healthy individual. Then moving quickly to glucose-responsive insulins, there are really two strategies that could be taken, and I know future speakers today will also talk about that. One strategy is to leave absorption from the subcutaneous tissue non-glucose dependent, but then followed by an glucose-dependent activation step in the bloodstream. That's the kind of pink or magenta color to the left. And to the right, you see another strategy where we get the glucose dependence in the absorption phase, and then a non-glucose dependent action profile in the bloodstream. And we prefer the magenta kind of principle where you get the activation or deactivation in a reversible fashion in the bloodstream, because that will be quicker in also reversing insulin action and getting the insulin back in an inactive depot once blood glucose is going down. So those strategies can be pursued, and we've done both. We've also acquired a small company in Bristol for a major sum of money a couple of years ago, actually 5 years ago almost, that worked on chemical glucose sensors that could be coupled to the insulin molecule to make the insulin action glucose dependent. And the idea being that an insulin molecule here in the kind of bluish color in the top there would be equipped with both a glucose sensor, that's then the glucose bound, the microcycle, that was the one licensed from Silo, and a glucose mimetic, that's the glucoside to the right with a starish-like appearance. And at high levels of glucose, the glucose would be bound to the macrocycle, and that would leave the insulin molecule in an open form, and would be amenable for receptor recognition and biological action. Whereas when glucose concentration goes down in the right panel, there will be a binding between the glucoside and the macrocycle, both sitting at the insulin molecule, that would bring the insulin molecule into a closed format that would not allow a receptor recognition. But it will allow these two forms to shift between an active conformation and an inactive conformation in an equilibrium, depending on the glucose concentration, and therefore both on rate and off rate could be controlled by a dynamic glucose range, in a dynamic range based on glucose concentrations. So that's the principle that we are currently looking at. We have embarked on both preclinical and clinical assessments of such insulins. Here you see that we've designed such glucose-sensitive insulins that has glucose-dependent receptor binding profiles, as you see to the left there, in the right concentration range with between 0 and 20 millimolar glucose, and also for downstream activation parameters, downstream of the insulin receptor, we see glucose-dependent action. And even in an animal model, like in the pig model, as you see here, when the glucose-sensitive insulin in blue is administered at time 0, it would be active in clearing both postprandial glucose to some extent at time 0, then giving basal insulin action between time like 6 to 8 hours and 24 hours. And then following a second meal challenge 24 hours later, it would still be circulating this glucose-sensitive insulin, and still when glucose clear at least partly a postprandial peak of glucose, which tells us that we have something there sitting and waiting until glucose goes up before it elicits its activity. So highly interesting, and of course promising for further clinical development of such insulins. That leads me to the last slide of my presentation. We've come a long way during the 100 years that we've known of insulin and the insulin molecule. We've learned a lot in developing novel preparations, both at the molecular level and at the formulation level. And insulin has also taught us a lot on basic science, actually, in just understanding what a protein is and how we can work with proteins for pharmacological action. And there's still a handful or more of opportunities to come for the benefit of patients with type 2 or even with type 1 diabetes. Thank you very much for your attention. And this was a little more than 20 minutes, but I hope we have a little time for questions as well. Thank you.
Thank you very much, Peter, for a wonderful overview and a great way to start this symposium. I can't see any other questions, but I've got a couple of questions that I want to ask you about. And it relates really to clinical practice and also my participation in an NIH hybrid closed loop meeting last week at Bethesda on behalf of NIH. And we talked about how much time in range is progressing. This is treatment which is, as you know, going to be standard of care in many countries, at least those where it can be afforded in the next few years for people with type 1 diabetes. But the solutions are not there yet, which of course is why it's called hybrid closed loop, as you know. And it occurred to me, as you were talking, that one of the issues, of course, is a rapid onset of action. But when I was listening to a presentation from Mike Riddell on exercise and the challenges, he pointed out that actually for those using hybrid closed loop, the problem is a cessation of action of quick acting insulin pumps. And you talked about rapid acting, and I just wondered, where's the biggest challenge? Of course, you can't provide details of the very early development of the insulins, but is the biggest challenge in increasing the rapidity of onset of action? And that's relevant to MDI, of course, because we ask patients to leave half an hour gap between injecting and eating their quick-acting insulin if they're using multiple injections. But it occurs to me in hybrid closed loop, arguably the biggest problem is cessation of action as quickly as possible, which is, of course, so difficult and relevant when you're engaged in aerobic exercise. And I wondered if you'd comment.
I think I think they go hand in hand, Simon, because the way to to control, you could say, the offset of action is often with insulins that are given subcutaneous, which is also the true for the pump systems, dictated or governed by the rate of uptake. So for most insulins, we have what we call flip-flop kinetics, and that means that what governs the offset of action is really the absorption rate, because the inherent, if you give an insulin analog IV, you have a very quick offset of action in actually a few minutes, and that gives you the kind of the true, so to speak, inherent rate of elimination of an insulin. But when you give it subcutaneously, your offset of action seems to become longer, but that's because it's the uptake of action that's governing also the offset of action. So the way forward for an ultra rapid acting insulin for subcutaneous pump use is to make it the onset of action as quick as possible, and that will then also spill over to a quick offset of action.
Okay, thank you. We've had one question, and I apologize if I pronounced the name of the questioner. H. Nathan, it says. I don't know if that's one name or two. And the final question before we move on to the next speaker is, out of all the promising developments that you've seen and some of the papers that you quote now a few years ago, so it emphasizes the challenges. If you were to say which is the most exciting, and you've been in the business a long, long time, what would you pick?
First of all, I can say that I've been surprised what proved to be possible when you aim high and are ambitious with respect to what you set out to accomplish. So had someone asked me, can you do a basal oral insulin like 10 years ago, I would say, well, it's probably impossible. But when we actually did it, it was not good enough for clinical utilization, but it was possible. I think I'm very excited about a once-weekly insulin because it will be realized. It will come out there, right? So that's, of course, for someone like me who is in the business of the pharma industry, I mean, we are really here to bring something really out to the patients. And that excites me quite a lot, that we can cut the number of injections so much for basal insulin. But if you ask a question, what do you think is most exciting from a concept point of view, then this holy grail of a glucose-sensitive insulin is phenomenal. I mean, if we could make an insulin that is perfectly depending on glucose concentration in the right range, so it's inactive below 5 millimolar and fully active above, like with a square wave kind of onset of action profile, it would be phenomenal. And I would say, never say never. It may be possible. We've had the first one in clinical trials. It was not good enough, but it did show glucose-dependent action. And we are working very dedicated to try to make that happen for the future.
Great, thank you very much. And there is one other question, but I'm afraid we must move on. So I'd now like to ask Professor Reinhard Becker, who's again one of our members of our scientific advisory board. Thanks to him and colleagues not only for participating in the discussions, but joining the symposium today. I'd like Reinhard to introduce the next speaker. Over to you, Reinhard.
Thank you, Simon. I hope everybody can hear me. So, dear colleagues, thanks to Simon and the group, and on behalf of this little advisory subgroup who's trying to match the intent of the foundation with the scientific challenges just laid out by Dr. Kurtzhals for the diabetes treatment, I have here the privilege to welcome Dr. Gu. In his current appointment, Dr. Gu is Professor and Dean of the College of Pharmaceutical Sciences at Zhejiang University in Hangzhou, China. But if I may say, actually, given the various appointments and activities you occupied with while staying in the United States, you're a role model of scientific exchange between People's Republic of China and the United States. And this includes some entrepreneurship. Over and above, you're also endowed with the perseverance required in pursuing the goal of glucose-responsive smart insulin and delivery patches. So thank you for sharing the experience in this matter. And if I may say, then for the upcoming 20-25 minutes, the screen is yours. So welcome.
Yeah, for Professors, Becker's introduction, and thanks so much for JDF UK for organizing such a fabulous symposium. And it's terrific to see many friends here. So I will briefly introduce our work associated with glucose-responsive insulin and the delivery patch. So our lab actually very interested in bio-inspired design for drug delivery systems. So typically with 2 tracks, one way is like to directly apply the live cells for drug delivery, especially to leverage their targeting capability as well as the cargo release behavior to enhance the delivery performance. Especially we are interested in using the platelets as a carrier to deliver drug, especially for targeting inflammation sites, the bleeding sites, for inhibit the tumor recurrence after surgery, also directly targeted to the metastasis. And we just actually initiated the IIT test using the platelets, which actually conjugated with the IL-2 to form the platelet drug conjugate for treating metastasis. So the other way is apply the materials like a polymer, liposome lipids to mimic the function of the live cells to deliver drug at the right time, right places, especially interested in the glucose-responsive insulin delivery. So just some background slides, I just want to jump. Especially want to mimic this kind of, the tighter relationship between the insulin secretion and the glucose levels. So as you know that for the drug delivery systems, typically, for the synthetic artificial pancreas involving 2 key elements. One is the factor that can sense the glucose level, but not directly actually provide the number, the digital, but actually, let's say, to pass the signals to the materials or to the insulin and to activate them to either release insulin or directly activate the insulin to reduce the glucose level, and then just follow the closed loop release system. So in the past, over 40 years, actually many pioneers in the drug delivery systems interested in this field, including Bob Langer, who is also speaker today, and also my postdoc advisor, and especially also many big farmers, including Novo Nordisk, certainly, and Merck, Sanofi, Lilly actually was interested in this field. And also in the past 10 years, many startup companies launched, including the Novis, actually our own startup company, and Protomer, and in Los Angeles, and Zello in Europe. So just some brief background, right? I think many people already know that, so just jump. And in 2015, we reported the first prototype about the glucose responsive smart insulin patch. The basic idea is to integrate the glucose responsive insulin delivery system together with the transdermal polymeric microneedle array patch. So this is our first design, especially associated with this enzymatic reaction. The glucose oxidase can catalyze the glucose to gluconic acid, and meanwhile reduce the oxygen level, okay, and also generate the hydrogen peroxide. So actually since 1980, many people are interested in leveraging the reduced pH to perform the glucose responsive insulin release. And however, actually it's a bit challenging to quickly reduce the pH level in vivo. So then we further like look for another way, a new method, like for example, to change the response mechanism to potentially enhance the insulin delivery or like quickly reduce the insulin. And then we focused on the generation of the hypoxic environments. So especially then design such hypoxic responsive material. So simply modify the natural immunome group to hyaluronic acid, okay? And the hyaluronic acid is like water soluble, and this part is like fat soluble. So this is actually amphibitic structure. So in the water, they can self-assemble into like a micelle or like a vascular-like structure to encapsulate the insulin, as well as the enzyme inside. And once the glucose level goes up, the oxygen level reduced, and this one will undergo a bioreduction process. The nitric group will become like amine group, and then the whole structure is like a more water soluble, and then can be dissociated, the complex structure, and insulin can be released. And suddenly, the glucose oxidase has like a relatively larger molecular weight, so relatively, released like slowly, but the insulin is like can be quickly released. Then once blood sugar level goes down, less insulin released, or just like a basal release. And to achieve the EZO treatment, we further embed such vascular or micelle-like structure into the microneedle array patch. The height or the length of each microneedle is about 600 to 900 micrometer. So the most nerve cells actually is like around like 1 millimeter or 1,000 micrometer away from the skin. So that's why we call it like a painless treatment method, because the microneedle actually can't interact with the most of nerve cells that significantly. However, you can still feel the pain. It's not a no pain. We just call it like a painless treatment. And the glucose can come in, and the released insulin can go out, okay, and it can reduce the blood sugar level. So this is actually our first design. And we actually applied the motor-based method to form the microneedle array patch. We pour this kind of solution with hyaluronic acid together with the formulation, and after drying it, we can shine a light to perform the photopolymerization. Actually, we certainly, with some like a modification to modify some double bond to the hyaluronic acid, and then you can cross-link it, and eventually you can form such like a uniform structure with the insulin formulation encapsulated. And once we apply such a patch to the mice, they can quickly reduce the blood sugar level. You can see the black line, and it can maintain in the normal range, like below 200 milligram per deciliter for a while, then increase. Okay, for the control group, like without the glucose sensing enzyme, and it quickly went down and quickly went up. So you can see the difference. And certainly you can also adjust the enzyme level as well as the insulin level to achieve the potentially personalized medication. And we also performed this study. We put one patch on, blood sugar level went down. And at this time point, you put another patch on. And for the smart insulin patch, actually, you can see the red line can still like maintain the blood sugar level around 200 like for a while, then increase. But for the patch with the pure insulin inside, without the glucose sensing elements, then the blood sugar level can further go down and even to achieve the hypoglycemia. So I think, we quite a smart insulin patch not only can respond to the high blood sugar level to reduce the blood sugar level, and more importantly, can also like reduce the risk of the hypoglycemia. And we further like summarize some like criteria regarding the translation of such patch. For example, actually in our group, we perform this kind of study in parallel, like with different students, postdocs, and we really want to, bring the most like exciting, most feasible methods forward for the clinical side. So such criteria are actually very important. For example, with the high loading capacity, high bioavailability of the insulin, but still like with a small size, and excellent biocompatibility with only insulin released, but not the material, because such patch will be used like every day. And also for the manufacturing should be fast, should be with low cost, like a scalable, process. And then we actually designed different methods, strategies to achieve the glucose response mechanism, because the first prototype is based on the enzyme, but later on we found that somehow they can cause the inflammation, partially due to the generation of the hydrogen peroxide. So then we also test on the PBA, phenylbutyronic acid based one. But for this system, how to avoid the burst release, especially for the hydrogel based formulation, is very important. So actually, since like 2018, 2017, we developed some like a new method, especially to form such so-called charge switchable smart insulin complex to mix the phenylbutyronic acid based polymer together with the insulin, and based on the electrostatic force, they can form such complex. Okay, it's not like a gel-based formulation. This is just like somehow like just a powder. Okay, and you can see here we, applied such a component. This here is like the furan-based group here, especially to enhance the response behavior and to modify it to the polymer chain. And here's like with amine group. Okay, so they have this kind of certain affinity toward the negative charged insulin to form the complex. And glucose actually can change the charge status of such a complex, because it can interact with the phenylbutyronic acid. Okay, and from uncharged status to the negative charged status. And then once brush level goes high, somehow the more positive charge status can be weakened, and the affinity between the insulin and the polymer can be decreased, and the insulin can be raised outside quickly from the complex. And this is actually really exciting results. Actually, we validated that such glucose response behavior can be clearly demonstrated, from the in vivo. Here's like a mouse study. Okay, this is the glucose tolerance test. Once the blood level becomes normal, we like gave like a shot of the glucose high level, right? It's like a 3 grams per kilogram, and then quickly we detected the increased level of the insulin from the plasma. Okay, this is actually a very clear in vivo glucose responsive insulin release results, and we repeated it for many times, and it's like really reliable. And further, we like test different polymer. For example, we here's like also apply the polylysine as the backbone, and then modify the phenylbutyronic acid to it. And similarly, we can achieve very clear glucose responsive insulin release behavior. This is in vitro, but you can see the difference is really significant. It's like around 10 times difference here compared to here's like the highest one is like 400 milligram per deciliter, and here's like around 100, 200 milligram per deciliter. The difference is like very significant. And similarly, also for the glucose tolerance test, we can also achieve the clear glucose responsive insulin release results. Unpublished data, this is another similar mechanism, but different materials under review currently. And we call it like a charge also switchable complex, but very importantly, it can last a longer time. This is for the mouse study. It's like can maintain blood level like below 200. It's like around like 200 hours. And for the pig, we increase the amount of the insulin. Certainly, it's not linear, right? It's like for 25 kilogram or 30 kilogram, pig, we just inject like a 4 to 5 milligram insulin to it. It can still like maintain the blood sugar level in the normal range for up to 1 week. So this is actually really exciting. That means not only can achieve the long-acting insulin release, but also its glucose responsive can reduce the hypoglycemia significantly. I mean the risk. Okay, and also very importantly, they can't form the fibrosis. Okay, so it can gradually absorbed, by the body, and certainly partially due to the such formulations like more like a network based. Okay, it's not like a bulk material or hydrogel injected inside. So this is actually also quite interesting. And back to the smart insulin patch, we further like in 2015 we launched a company like Genomics, and then we can test the different system, different materials in the patch, move forward. And currently, quarter-sized smart insulin patch could effectively regulate the blood sugar level of 25 kilogram type 1 diabetic pigs in a normal range for up to 24 hours. So this is actually really clinical relevant. So that means for 60 kilogram or 70 kilogram people, and 2 to 3 patches can potentially maintain the blood sugar level for 1 day. So that means can potentially replace the subcutaneous injection of the insulin. And the mechanism here is also phenylbutyronic acid based. Okay, so we actually identified a group of 4 different monomers, 2 monomers with amine groups. Like here, just also, they have like a high affinity toward the insulin, relatively negative charge. And this is 2 double bonds can form the cross-linking, network. And this one is phenylbutyronic acid. So very importantly, such solvent can well distribute the insulin inside without any water. Okay, if you add water to it, this could be a hydrogel, right? And can potentially form this kind of burst release, or you need to wait for a while until drying. So then, you can just pour such solution, the mixture, to the mold and shine the light to it. And certainly with some like a small amount of the photo initiated. And quickly we can form the microneedle array patch, okay? Just like the resin, to fill your, like the teeth hole, something like that, right? The dentist sometimes like shine the light to it, and actually to form the resin. And here is like for the microneedles. All right, so since now the whole structure is glucose responsive, so then the loading capacity is quite high, also with the high bioavailability. And also the administration, again, just like the other microneedle patch, very easy. And very importantly, you can stop the treatment at any time. If you feel uncomfortable, you can just detach it and throw it away. Not the oral insulin, right? Or not the subcutaneous injected insulin, right? Once inside, it's harder to get it out. And for the patch, you can stop anytime. And also, this is a cold chain free system, because the cross-linking structure actually can stabilize the the structure of the insulin, and it can maintain a longer time regarding the stability and activity. So on the room temperature, we just put it there, okay, on the room temperature, and but on a drying environment for like 1 year, and we get it out compared to the freshly made smart insulin patch. Regarding the insulin activity, bioavailability, almost the same. So this is actually also one advantage. And for manufacturing now, it's also very easy. And we put the patch on. Here's like for the large animal study, and after 2 feedings, around 20 hours went up. And for the control group, without the glucose responsive elements, 1 feeding, it's up. But here's like you can see, due to the detection limit, because here's like just use the Medtronics, a sensor. So under 40 milligram per deciliter, we can't detect the signal. However, we just draw the blood from the pigs and to test if with any like hypoglycemia. and actually the pigs actually just keep a normal glucose level actually without the hypoglycemia like over here's like 2 days, and we even last like over 1 week. No any hypoglycemia happen. And once you get the patch out, you can see the structure is still there because it's cross-linked. Only insulin go inside, okay? And we can repeat it. This morning, put one patch on, tomorrow another patch, and it can last like over 40 hours with limited inflammation side effects toward to the skin. And also for the glucose tolerance test on pig, you can see with the blood sugar level goes up, the release of insulin also went up, okay? Very clear in vivo glucose responsive insulin release. And we just initiated the IAT test also in Zhejiang People Normal Hospital in Hangzhou. And we also developed the dual self-regulated glucose responsive patch. We put the glucagon inside, okay, and form the hybrid patch. And here, due to the difference of the PI value between the insulin and the glucagon, we can adjust the ratios of the monomers, okay, and we can later on achieve like on the high glucose level, insulin can be released from the red parts of the patch, and once blood sugar level becomes too low, and the glucagon can be released from the 1/4 of this patch, and just mimic the function of the alpha cells. Okay, due to time limit, I just simply pass these results. And we can even like further screen the monomers and put the insulin and the monomer, insulin and glucagon together into the material. But here we modify the glucagon a bit, like with 3 arginine, we modify to the glucagon and further adjust the PI value of the glucagon, and we can use the same material to achieve the operated release of the 2 different proteins. It just passes these kind of results. Actually, this is with high glucose level, insulin can be released, low glucose level, glucagon can be released. And this is in vivo results, but due to time limit, I just pass. This is also another interesting work, so-called UAV mediated targeted drug delivery for first aid. We actually combine such a glucagon patch, for example, to the UAV system, okay, and they can identify the certain people, okay, but here's like 2 single people, okay, by the way, and then automatically put here's the such device to, for example, here's pig, and it can trigger, here's the red one's trigger, and then push the patch to the skin, and then can quickly release the glucagon. So this can be used for the first aid. Okay, but here's a movie, just simply. Okay, I just pass. Okay, and this is actually also platform technology. Okay, so for the different applications, due to time limit, I just pass. Very importantly, we also run companies called Microzine Farmer. For this company, we especially build up the large manufacturing pipeline for the microneedle fabrication manufacturing. Also for the microneedle for the mRNA lipid nanoparticle delivery. Last story, very importantly, just give me a few minutes. So we conjugated the glucose transporter inhibitor to the insulin to achieve the smart insulin delivery. So glucose transporter inhibitor actually can bind to the glucose transporter and to increase blood sugar level. And the insulin can bind to the insulin receptor to reduce blood sugar level. And the one large molecule, one small molecule, we bind them together with the operator functions. And we found that this is very interesting. We can make the insulin smart itself. So once blood sugar level goes up, such we call the I-insulin. This is actually one kind of glucose transporter inhibitor, especially can target it to the GLUT4. And then, glucose can competitively enter the cell to detach the I-insulin to make the balance more to the insulin receptor to quickly reduce the blood sugar level. Okay, and also very importantly, overdose injection of such insulin won't cause the hypoglycemia. For one kind of our such insulin, we even increase the amount of the insulin to like almost 20 times, 20 times larger compared to the one dose of insulin. Still can't cause the hypoglycemia because they can block the glucose transporter. Sometimes overdose even can increase the blood sugar level a bit. This is also very exciting recent results. 2 milligrams of such insulin can maintain the blood sugar level of the 30 kilogram peak in the normal range for up to at least 1 week, sometimes even like 10 days. So not only long-term, maintain blood sugar level, long-acting, but also reduce the risk of the, hypoglycemia. Some other results I just quickly jump. And by the way, this is my car license plate, deliver insulin kind of. With that, I would like to thank my team and the support from funding sources. Thank you.
Thank you very much, Ben, for this really firework on plethora of ideas you presented to us. And I was picked here because I was in clinical pharmacology all the time in a big company, as Sanofi mentioned, with a little bit background of chemistry. But what you presented really requires a very careful study and really to grasp each and everything you just prepared. You have shown a lot of ideas. I have one question when it comes to the animal models, where you want to show all the activities. You picked mice, and as far as I know, this and you look for the epidermis injection of the patches. I recall that even in dogs, you're misled by the different skin structure compared to the humans. The pig is different. You learned it from Peter Kurtzhals as well. The pig is different when it comes to absorption kinetics, but the other animals, the mouse and those, is a bit misleading. And I understand that you will go into the pig later on with all your approaches. Is it correct?
Yes, for the mouse study, actually, the STD induced the mouse, and this is STD induced type 1 diabetic mice. And your question is like for the pig study about what? Can you repeat your question?
My question was only the various approaches you presented to us. I think you're going to test certainly in different animal species because that can be crucial when it comes to the pharmacokinetics. But,
yes.
For the sake of time, I just received one question here. It is about, do you have any idea about how frequently the patch would need to be replaced by somebody else, and how long would it be last according to your suggestions in a child?
Yes, so for the back to your the first question is like to the pig study. Actually, the pig skin regarding the structure is quite similar to the human. So for the patch study, usually we put on the the pigs, for the microneedle-based study, and we can maintain the patch on the skin like for several hours, for example, 2 to 3 hours. And for some cases, we can put the patch on like for over 1 day, and we found that there's like a limited side effects toward the skin, just sometimes a bit red, but after that, they can, disappear within a short period of time. Yeah.
Okay. So thank you very much that you took the time. It's late in the area in Hangzhou, and I will hand over now to Simon again to continue with the session. Thank you very much for taking your time for the presentation.
Thank you very much to both of you for a wonderful talk. And we now must move on. Just before I do, I just want to point out that we're also hoping to write a review involving all these speakers, which we will publish in a journal as a record of this meeting. So I now want to move to the next speaker, who's Professor Chou. He is an assistant professor of pediatrics in Stanford. He's worked in Harvard, MIT, with some great researchers, and he's going to talk to us today on molecular engineering of insulin progresses and challenges. Danny, over to you, and many thanks for participating in this.
All right. Thank you very much for the organizers for the invitation, and thank you very much for your participation. My name is Danny Chou. I am an assistant professor of pediatrics endocrinology here at Stanford. Just to let you guys know a little bit, so this is 6 a.m.-ish in our time. So possibly throughout my talk, my young son may wake up, and then I just hope that he will not destroy the presentation, but just to let you know that there's a slight possibility that I may need to deal with him a little bit. So today it's my great pleasure to talk to you about some of the insulin work we are doing, and then I title it molecular engineering. So this is basically we are making insulin analogs that can potentially address to some of the very important challenges we try to address type 1 diabetes. And then I'm actually quite happy about the first presentation in the way that that's sort of a very, very good background about what we are going to talk about. And then hopefully this will resonate quite well with most of you here. All right, so just a brief background in the way that basically what we are trying to do is either to make things that's even rapid acting or something that's long acting, but give you more glucose sensing or smart properties. And then the idea of the majority of my talk will be surrounding around how to make insulin to be potentially more rapid acting by making it more monomeric, and then also use that as a concept to jump into the glucose sensing part. This is really the reason why we step into it, and I think the first presentation also talked about some of the critical challenges here. So back at the time when I first started my group, actually one of the very first trials using the closed loop system just got out, and then you can see that using the kind of the so-called artificial pancreas device, the time now people with type 1 diabetes that can spend within the range that people care about, it's much better. And then this is really a terrific news. And then what I find in this New England Journal of Medicine paper in the very last paragraph is they still say that the currently available rapid-acting insulin analogs still have relatively slow absorption after subcutaneous injections. And basically, this is one of the reasons why we started to try to find a way to address this challenge using molecular insulin engineering. This is a study from Tim Heise's group in the way that they are basically performing some human studies in a way try to see how fast the ultra-rapid insulin Lispro and Fiasp can do compared to their original insulin analogs from Lilly and Novo. So you can see here, it's a terrific formulation in the way that using the excipients they added, they can basically achieve much faster onset action compared to the original molecule. And then one would argue that probably the offset, or basically the off part, is closer to the original molecule compared to the onset part. But still, this is making the pump decision a bit easier in the way that now that the wait time is a little bit shorter. And one may argue how to address the offset part in a way, also try to see bigger and bigger delta there. The way we think about is really related to the insulin maximization that previous talks talk about, in a way that basically when you are doing subcutaneous injection, when you have something bigger, people will get absorbed there slowly, and when you have something smaller, it could be faster. But really the challenge is insulin is a hexamer for a reason, in a way that is the way we try to make it much more stable, because our body makes lots of insulin, and small things tend not to be that stable, but by forming in hexamer, it's basically a terrific storage form for beta cells to keep it. So it's there for a reason, and then the insulin sequence was designed such that basically the last few amino acids in the B chain plays a key role in the dimer formation situation. So if one go to check the crystal structure of an insulin dimer, you will see that there is a very strong anti-parallel beta strain interaction here. And then one of the initial thought we have is, can we actually remove this part to make something that's monomeric? The answer is yes, it probably is monomeric, but people know that for a long time that this is also the same region responsible for a very key part of insulin receptor binding. So you can remove it, potentially remove the dimer formation, but you will also remove the receptor binding affinity. So this is the reason why this is kind of a dilemma in the way that you kind of need that part for receptor binding. So it's pretty hard to get rid of that part for making these only monomeric but not dimeric and the hexameric. So this is a video about a corn snail trying to eat fish. And this normally doesn't make a lot of sense because fish should have been swimming away such that corn snail cannot eat it. But in this case, you will see that the snail just opened the mouth and then eat a fish. So there must be something going on there. And then exactly, there's something going on there. And then this is from my former colleagues, Professor Helena Safavi and Professor Toto Oliveira at Utah's originally in biology department. Basically, they study what's inside the corn snail venom. So basically what happened is the corn snails secrete venom such that they can intoxicate fish. Mostly they will find a lot of things like neurotoxins or ion channel blockers, which makes perfect sense because those are agents that can make organisms not to move. But in some rare times, like in this paper in 2015, they found a molecule looked like this. And then of us here who are familiar with insulin, you will know that this actually have the same disulfide skeleton like insulin here. And then what is even more special is it actually doesn't have the dimer formation region here. So this is part of the reason why we decided to work together in a way that to see there are some features we can learn from the venom insulin such that that can help us to design a monomeric insulin. And potentially down the road, that can be something that's much faster to be absorbed to the body and then potentially also have a faster off rate as well. This is to use the analytical ultracentrifugation to basically measure the association state. As you can see that the association state is 1.05. So that means that this molecule is pretty much a monomeric. And then the thing what we are really interested in originally is to see whether this molecule is really something that can be targeted towards human insulin receptor. The answer is not quite. So it's somewhere around 20 to 30-fold weaker in both binding affinity and potency compared to native human insulin. But it's still quite interesting because DOI, or this octapeptide insulin, I show you that it's somewhere around 500 to 1,000-fold weaker in the binding affinity. So this suggests that there's something or some mutations in the venom insulin for us to study in a way that could be helpful to do the molecular engineering. Long story short, in 2016, our team with collaborators resolved the crystal structure of the venom insulin. And what we believe in this case is basically, although they don't have the same B chain C terminal, which basically a very important part of the interaction is use this B24 phenylalanine to bind to a hydrophobic pocket of the receptor. What we believe at that time is potentially B15 and B20 both got mutated, such that, the original molecule was leucine and glycine here, but both got mutated to tyrosine such that they could potentially compensate for the mission role of this residue here. And then long story short, because it's already published in 2020, but basically what we end up having is a molecule we can cut off the final 8 amino acids. So basically this is the kind of the human insulin backbone in a way that could be monomeric, but it has much lower activity. But if we put in 4 mutations, 2 on A chain, 2 on B chain, exactly learned from the venom insulin sequence, we can actually make an insulin that's pretty much the same potency as human native insulin, but while maintaining the monomeric state. And then this is a business figure, but just to show you that they behave quite similar to native insulin. And all 4 mutations are important. If you remove one out of the 4, you lose somewhere between 4 to 10-fold in binding affinity. What we believe in this case is, in addition to the tyrosine B20 mutations that we learned from the structure to compensate for the role of the missing B24 phenylalanine, what we believe that there are 3 other sub-bridges created by the snail venom insulin mutation, such that it increased the affinity with the receptor, such that now with all 4 mutations, now the affinity is quite similar to the original molecule. After that, our collaborators find even more venom insulin sequences, and then there's one class that really caught our eye in a way that it actually doesn't use the same mutations we just found out about, 2 on B chain, 2 on A chain, but instead it has a longer A chain. So we thought that this is something quite interesting for us to follow up, and because of the previous experiences on the engineering, now we know that potentially we can still cut the B chain off and then just try to study what an extended A chain would do, and it is exactly how we did it. So very quickly, by putting in all the different extended A chain sequences from the venom, and also doing some aniline scaling, we very quickly found out that this HALQ extension works really well in a way that basically it's achieving the same feature we are looking for, in a way that it doesn't have the dimer formation region on the B chain, but with the extended A chain, it can potentially do the job there. And then basically it's just some structural activity literature to show you that this HALQ molecule looks really well in a way that it has this basically the same binding affinity with insulin receptor compared to native human insulin. And this is with IGF-1 receptor in a way that same as human insulin, you actually have somewhere around 100-fold weaker in binding affinity. This is at a time when cryo-EM structures were widely used, so we were able to work with 2 terrific structural biologists, Chris Sale at University of Utah and Uno Coston in University of Dresden, in a way that we solve this insulin, we call it mini K1, in complex with human insulin receptors. So you can see that basically, like all the previous structure, there are 4 insulin bind to 2 side 1s and 2 side 2s of the receptor. But what we are really interested in is how does a molecule with this extended A chain really compensate for the effects of the missing B24 phenylalanine. What we found is actually in the molecule HALQ, that L in A23 residue, which is basically not there for native human insulin, because you would have stopped at the 21st amino acid. What it has is basically this A24 leucine occupy the exact same 3D location where the white one, which is the human insulin complex case, the white one, the B20 phenylalanine would have occupied in the 3D structure. So basically, this is the way that the snail venom potentially do evolution use this exact mutation with the right extension in the way that it will occupy the exact same location there, and then that's what we believe that's the case. If you are interested, basically, I mean, we are here for the insulin, but there are actually quite some interesting studies of how nature used to repurpose hormones to do some of this. And some of you may be very familiar with the case of exenatide in monster for the case of GLP-1 analog. So last year we wrote a review along with Helena to kind of review some of these repurposed hormones. So if you're interested, you can you can try to read this one. I suggest to you that basically for the extended A chain one is using the longer helix to occupy for the same space. At that time, we start to think about, because of the longer A chain, potentially the extended part could be quite close to the B22, 23-ish, this region of the insulin. We do some measurement of the distance between the C alpha carbon, and what we found is actually it's quite close in the way that potentially one could think about a disulfide bond there. And I will tell you why we have a disulfide bond there. But originally, what we just try to do is try to engineer a fourth one there. One of it could be something that could be more stable, but we do some engineering in a way that we actually found that if you do something that's somewhere around A22, B22, so basically B22 would have been an origin. We mutated to a cysteine and then introduce another cysteine on top of the insulin agent. We can find a molecule in cell culture. It basically have the same potency to activate a receptor. In mice, we can see that they basically have the same glucose storing potential as native insulin. We saw the structure of this 4-disulfide bond insulin and then compare with native human insulin structure. You can see that basically it has exactly the same backbone there. And then to have the 4-disulfide bond, all it takes is a little bit shift on the side chain degree, and then basically you can have something like this. But then as you can see that it doesn't really affect with this part of the region, so it can still have the conformational change, and that's why it's still active. The reason why we are interested in this is actually because, so this is a review written by Professor Michael Weiss group, who will give a presentation later. They talk about what are the key things one should think about trying to insert a glucose-dependent switches. And one of the obvious way will be basically the kind of the traditional thinking of a pseudo-pro insulin type of things, and then in a way that basically if one can have a linkage here and make it glucose dependent, that may lead to a glucose dependent insulin activation behavior. And then two other examples will include a B4A10 4-disulfide residues previously reported by scientists at Novo, and also the B22A22 stuff I just talked to you. This is a study the Weiss group published not too long ago in a way that basically they can design it in a way such that they can have the linkage in the pseudo pro insulin part I mentioned to you, in a way that you can see that they can actually achieve a pretty good insulin receptor activation with in the presence of the fructose, although not so much on the glucose part. But this is actually not their problem, because this is related to the original design of the phenylbaronic acid they use. And also, this is also from the same review they published, which is a terrific knowledge base for the whole stuff, in a way that basically, because baronic acid tend to like the furanols, which is basically the 5-member ring glucose. And unfortunately, this is only a tiny bit of the glucose population. So compared to the majority of the pyranose population, which is actually will be occupied by actually this kind of synthetic lactine stuff that the first presentation talked about. So basically, this is kind of the initial problem about a single baronic acid to recognize glucose. So potentially, we would need to have some sort of novel small molecule-based glucose sensors if you want to go for that. And then I'll argue there are probably two good examples of this to achieve something with a higher affinity. The first one is the Zylo Novo Nordisk one that they already talked about, so I'm not going to go deeper. The second one is actually a 5-boron in a way that potentially, because you have 2 baronic acid basically handed in the right geometry such that they can bind to a 6-member ring glucose quite well. And it's a way to increase affinity and also increase the selectivity toward glucose. So they talk about, I mean, the Novo one, they just talk about the design, but basically they were able to put both the macrocycle and the glucose site together in a way that try to basically make it a way that with or without glucose to compete for that, they can see different affinity with the glucose. We're actually trying to do something quite similar, but now you see the bidentate baronic acid, so the 2PVA1. This is a work in collaboration with my colleague Tosu Huang in bioengineering department here at Stanford. What we are trying to do is we try to design using computational protein design techniques to put 2 PBAs along with a glucose model in different part of the insulin. And the way we are trying to do is to get the structure from the receptor activation analogs in a way that we can now introduce this bidentate PBA in various parts of the receptor and then try to calculate what will be the right, the most stabilized conformation using computational approaches. Once we have this information, we will move back into see whether with or without glucose, they will have a different conformation. And then now we will put it back to the receptor such that we will see whether there's a clash or not. So ideally, what you try to find is if they are in the glucose binding mode, this whole analog should fit in very well into the insulin receptor structure. And then when there's no glucose, you would want them to have some sort of conformational change such that now the new conformation will make a clash with the receptor such that the new conformation now without the glucose will lead to much lower affinity and enhance fewer insulin activity. And this is a current work we are trying to do, and then hopefully in the not so distant future, we were able to publish some of the work in this area. And then finally, this is the people in our group. And then this is my email here. If you have any questions, then feel free to email me offline. And this is the funding support. Thank you very much for your attention today.
Thank you, Danny. That was a fascinating talk. I just want to ask you, I understand the idea of developing a PBA analog. Most of your work is in vitro, as I understand it. The collaborators that you're working with, will they be the ones to take this into in vivo studies? Where actually are you? And do you have the capacity in your own lab to do this, or will you be reaching out to others? And I suppose the $50,000 million question is, how long might it take you to do that?
Yes. So, I mean, of course, we're in academic institutions in terms of the drug development perspective. This is not really what we are super good at doing, although we do have quite some work related to the in vivo part. And because just the time constraint, I didn't talk deep about that. So we perform my studies, rat studies, and even pig studies in-house here at Stanford in the way that we have the capacity to do that. Although I will say our capacity is relatively limited to just the simple insulin tolerance test and glucose tolerance test in the way that we inject the stuff in. We can see the glucose levels, and then we collect the blood to see the PK of the insulin. We have a collaborator, Professor Simon Fisher at University of Kentucky. They perform glucose clamp studies for us using rats, and that's basically the capacity we have. For something more than that, and potentially one day, if we can clear the toxicity part, one day we could move to clinical studies. You probably can see that I'm in the pediatrics and in the division. So there's quite some of my colleagues, they are involved in various clinical studies, even in the closed loop part where some of them are doing some investigational studies on new insulin. So this is part of the reason why we moved here, because this makes the clinical collaboration to be much easier for us.
Great, thank you very much. I think this is a question directed at you from an Andy Chapman, and I certainly will put it to you. And maybe you could answer it. What about oxidative debarylation? Boric acids can also be unstable to proteinolysis. How will they be stable enough? Can you answer that question?
Yes, this is a terrific question. And then basically, this is probably more to the chemists in the room, but a lot of us who are chemists who work with the boric acid will know that the oxidative debarylation is something that's very prone and very easy to happen in a way that basically anyone working with phenylboric acid, if it's in a little bit basic conditions, it's very easily to form the phenol. So basically the boron will become, instead of a phenol with the BOH2, you will quickly just get phenol with the OH there. So this is something that's very easily to happen. In terms of the in vivo, this is actually relatively difficult to happen because we have done studies where we inject some bronchic acid labeled insulin in vivo. And then when we are trying to collect the blood and then try to see the PK using mass spec, we can actually still see the bronchic acid intact. So I would say that this is more of it in the vial or one or outside of the body, one will need to be very careful about how to handle. But in terms of adding in vivo, they are actually relatively stable. So I'll say in addition to our work, there's also, I don't think they are published, but some patent work from Novo's team in the way that they also make some molecules with boric acid in it, and they perform some in vivo studies. And what I believe is the boric acid is still stable in that sense. So stability is one concern. They should be good enough. And then if you want to ask about some long-term toxicity or long-term safety in vivo, I think that will be another consideration, and then potentially we'll need more studies to understand.
Great, thank you very much. I think we probably now need to move on. Just a word to say that if your questions haven't been answered, if you make it clear who you're directing them to in terms of the speaker, it may be that if the speaker is still in the symposium, then they can answer your questions directly. So I'd now like to move to Dr. Tim Heise, who's my colleague, worked with me jointly in the Novel Insulin Committee, and ask him to introduce the last speaker before we'll have a short break. Tim, over to you.
Thank you very much, Simon. So I think the next talk will really build on the one we just heard. So it's time to talk about challenges, of course, but also about potential solutions for glucose-responsive insulins. And it gives me great pleasure to welcome Matthew Weber. He is the Keating Crawford Collegiate Professor of Engineering and also an Associate Professor in the Department of Chemical and Biomolecular Engineering at the University of Notre Dame. He has received quite a number of awards, including the ADA Pathway Accelerator and the JDRF Career Development Award. And his research group is primarily interested in applying supramolecular principles, leveraging defined and rationally designed non-covalent interactions to improve biomaterials and drug delivery. So I think he is in an ideal position to talk about the molecular engineering of glucose-responsive insulins, and we look very much forward to your talk, Professor Weber. Over to you.
Yeah, great. Thank you for the invitation to be here and the nice introduction, and looking forward to sharing some of our work here with you today. It's great to follow two former MIT colleagues as well in the lab of Bob Langer and Dan Anderson, and clearly we got our start kind of in the same place, and so it's nice to see Jen and Danny as well here and follow them. Hopefully I can match their impact as well. So first, I'd like to start by thanking the people on the left who do the work. Those highlighted in red contributed directly to the work I'll show today. And on the right, I'd like to thank the people who fund the work, of course, JDRF, the reason we're here. And then also the Helmsley Charitable Trust has funded a lot of my diabetes work over the years, and actually was sort of the nucleus for Jen, Danny, and I all getting started in glucose-responsive insulins back in our time at MIT. And so I've been very grateful to the Helmsley Charitable Trust for 10 plus years of support in this work. And so I don't think I need to convince people here that we still have a glucose control problem. This is relatively old data, but this was from a dual hormone pump study where this pump was supposed to be smart and kind of learn behaviors and adjust dosing accordingly. And this was several days after this learning was supposed to have taken place. And you still see this undesirable excursions in both the high and the low direction in terms of blood glucose. And we know that there's a number of challenges that can arise from this suboptimal control. Things that occur acutely, right? So, very, very scary hypoglycemic episodes, seizures, coma, death, these kinds of things. But then also, very long-term risk for increased risk of cardiovascular disease, stroke, other sorts of things that we'd like to also avoid if we're going to be treating someone for a lifetime in managing their diabetes. And so where do engineers get involved in this space? I'm a chemical engineer. I design materials. And so where could we get involved? Well, we could think about engaging with, cell replacement therapies and designing better materials for protection of insulin secreting glucose sensing cell populations. We could think about getting involved in some of the hardware or maybe mitigating surface interactions of pumps or sensors or other sorts of things like that may make life easier on diabetics, what we've kind of termed the bionic pancreas approach. Or we could get involved in the area of smart drug delivery, which would be the fully synthetic pancreas idea that my colleague Zhen Gu introduced just a couple of talks ago. This idea that we could introduce glucose sensing functionality into some drug delivery device or into insulin itself in order to facilitate enhanced function or glucose tunable potency or glucose tunable activity or bioavailability. And so this is the area where we've tended to get involved over the years. Danny and Jim both introduced this chemistry briefly, this dynamic covalent bonding that occurs between an arylboronate, a phenylboronic acid, and a cis-1,2 or cis-1,3 diol, such as glucose. And so we got started with this chemistry back in 2015, actually working with Danny in a paper we reported in PNAS, where we took insulin, we modified it to the B29 lysine residue, same site that insulin detamir is modified, with these aliphatic arylboronate conjugates. The idea being maybe we could hijack albumin binding, but do so in a glucose dependent manner. And so these conjugates could bind glucose, alter the hydrophilicity of the conjugate, and then therefore change the function. And we were able to demonstrate something that looked a little bit like glucose responsive insulin, right? I don't think we ever formally showed exactly what we wanted to show, but we could inject a single dose of this at time 0. We could simulate sort of a meals over the course of a day at 4, 7, and 10 hours by doing an intraperitoneal glucose tolerance test. And we could see that certain of these modified insulins were able to recover their properties fully in correcting blood glucose, even out to 13 hours or so after treatment. And so this was generally exciting. Working with Danny, who's a phenomenal chemist and great at modifying insulin, was awesome because he had to do the hard part, which was actually making and modifying the insulin, and delivering 8 milligram quantities of a purified modified insulin at the end of these studies. And so thinking about that, I started thinking, well, maybe covalent modification of insulin isn't really a great idea. Maybe we need to learn some things about formulation and other sorts of things to play with insulin properties without touching the insulin itself. And so one of the things we did, and this is kind of just a brief vignette here, is we started thinking about how to formulate insulin without the need for covalent bonds. And one thing we could do is we could leverage, in the introduction, it was referenced that I like supramolecular chemistry. We could reference non-covalent bonding of insulin, in particular the B1 phenylalanine, using a macrocycle that could recognize that as a host guest complex. And doing so, we could actually do non-covalent modification and formulation of insulin to change its properties, developing things such as very shelf-stable insulins that with 100 days of stress aging don't lose their activity. We could do things like stabilize the rapid-acting insulin monomer variants or alter insulin pharmacokinetics non-covalently. Or we could even do things like make all new classes of drugs by enabling co-formulation of insulin and pramlintide together. Somebody probably needs to mute themselves there. I'm getting a little feedback.
Apologies, that's been done.
Yeah, thanks. And so we could think about making all new classes of drugs, right? We could co-formulate insulin and pramlintide together, and we could inject these in a single injection, whereas their solubility profiles didn't really allow them to be put in the same syringe otherwise. And so this demonstrates one way to use formulation-centered approaches to take unmodified insulin and control its properties, control its activity, and control its delivery in certain ways. Another way we could think about doing this is to take unmodified insulin and start to put it into materials that could do the glucose sensing piece of the arrangement. And so other work that kind of started when I was at MIT working with my colleague Volkan Yeselier, we decided to start making injectable hydrogels on the basis of forearm peg macromers and use this PBA diol recognition event to facilitate glucose-dependent cross-linking in these networks. And so the theory would say, well, as glucose goes up, you increase competition with those cross-links, the network swells and the bonds break, and the encapsulated insulin is released. Unfortunately, these materials didn't prove to be super sensitive for glucose recognition and glucose-dependent properties. And we got a nice paper out of this. We talked a little bit about their pH-dependent properties, and this paper has actually been cited very, very well. but it doesn't actually work that well in terms of delivering insulin. And so we wanted to think about other ways of using hydrogels and polymers to formulate insulin. And so one of the challenges that Professor Chou just mentioned here is that these phenylboronic acids aren't very good at binding glucose. They don't bind glucose with affinities that are really in the physiologically relevant concentration regime of normal blood glucose. You see binding constants on the order of, say, 10 inverse molar. And maybe if we think about blood glucose levels on the order of 10 millimolar, we're a couple of orders of magnitude off in terms of the binding affinity we'd like to see, right? Another thing they do is they bind to other analytes like fructose and lactate and other sorts of things in a way that is competitive with their glucose recognition and in some ways better, right? And so they bind fructose, for instance, 5 to 10 times better than they bind glucose. And so if we're going to try for a glucose responsive insulin, it probably shouldn't respond to other analogs. And so I give this example sometimes of lactate interference, right, with phenylboronic acid sensing. And you wouldn't want somebody who had one of these glucose responsive insulins on board who have just finished a marathon, they haven't eaten in 3-4 hours, and now all of a sudden they're getting a dump of activated insulin into their system with the spiking levels of lactate in the body. Right, this isn't when you want your insulin to turn on, right? And so I think we need to think about making this chemistry more specific. And so Danny introduced the diboronate system. We've kind of also been exploring these as a way to improve specificity of glucose responsive insulins and glucose responsive materials. In particular, going back to that hydrogel chemistry that Vulcan and I had developed, and actually start to think about using diboronates as crosslinkers instead. And so this was a report we put out last year, where we could actually take some of this chemistry and we could make it more specific to glucose by integrating it as a cross-linking motif in hydrogels. And so if we think about this prior work, right, we could compare our system and we could say, well, now we can improve the glucose responsiveness by using diboronate sensing of glucose. And we could actually then maybe limit the responsiveness to non-glucose analytes. And so this was the general idea of the system where we wanted to test this. We wanted to put it up against kind of a standard fluorophenylboronic acid here, kind of shown in this teal color, to kind of compare kind of what would be sort of the prior art in the field to this new design. And so first, if we look at the molecular scale and we start looking at binding affinities, that sort of standard in the field, that fluorophenylboronic acid binds glucose with 8.6 inverse molar based on our isothermal titration calorimetry measurement, whereas it binds fructose by these same measurements with 670 inverse molar. And so, upwards of 50 or so fold increase in the binding affinities for fructose versus glucose for this particular chemistry. Lactate as well is binding, on the order of, almost 10 times as much. And then the diol, this was sort of an interesting finding. I think this explains why Vulcan and I systems didn't work so well. That diol that we were using for cross-linking actually binds with, almost 5,000 inverse molar binding affinity to the phenylboronic acid. And so we were getting super high affinity binding of the diol and not so good affinity binding to the glucose that should be competing with that diol, right? When we switch to this diphenylboronic acid chemistry, we get a several order of magnitude increase in the binding affinity to glucose. Whereas the diol is actually about the same, and the fructose and the lactate are about the same. And so we're actually able to go from a molecule that is more specific towards fructose than glucose and switch that and actually make a molecule that is more specific to glucose than it is to fructose or lactate. This is a little bit of polymer chemistry, but this basically shows that in the polymer system, if we fit a dynamic phantom network model for the polymer system, we can actually see that the binding affinity of those crosslinks is about the same for our diphenylboronic acid system as to the fluorophenylboronic acid system, which is telling us that the bonding stoichiometry is the same, and that's not what's underlying the function here. We can then look at the glucose responsive effect. We do a lot of rheology in my group, and we can see that the mechanical properties of these gels decay pretty readily with application of glucose, whereas the phenylboronic acids, the traditional chemistries, don't show much change in their mechanical properties. This then translates to a greater delta in terms of the release of insulin from these systems compared to the phenylboronic acid. Now, I would argue there's still a lot of leakage. There's still slow response in these systems. There's still undesirable features that we'd like to engineer out. But generally, the phenylboronic acid chemistry makes more glucose responsive gels. It also is able to make more glucose-specific gels, right? And so we can start to add things like very high physiologically relevant concentrations of fructose and lactate, and we don't see a decay in the mechanical properties of these gels. They look like the standard gels, but if we add the glucose, they drop off pretty considerably. Whereas this system here has pretty comparable response to fructose, lactate, and glucose. Now, when we translate this to the release setting, what this means is when we modulate lactate levels, we don't see a difference in release. The difference really is specific to the glucose level, versus the phenylboronic acid system increases its release in response to both glucose and lactate, right? And so it's actually demonstrating kind of sensitivity to both analytes, which would be a problem in the context of a release study. We wanted to take this and test it in a mouse model, and we could look at kind of the responsiveness via area under the curve. And this system, in response to glucose tolerance testing, is able to respond to a couple of sequential challenges, even after several hours of being dosed, with pretty tight control following these challenges and a reasonably fast return to baseline levels. Another issue that we don't talk too much about, we call these gels in that original paper I mentioned, we call them injectable. And I've done the injections, and if you inject 20 or 30 mice with these things, your forearm gets pretty sore. You've got to push on that syringe pretty hard, right? And so that's not really a reasonable thing to ask someone to do. It's not really feasible for injecting gels in these pen type injectors, right? And so we wanted to actually think about a thermoresponsive mechanism for gelation. And this was actually the subject of the JDRF Career Development Award that was mentioned in the intro that we started writing about. Was thinking about making these gels more thermally responsive. Could we actually take and integrate this chemistry with a thermoresponsive micellar polymer? And so we were able to do this. We could get gels that form upon introduction to body temperature. And so you'd have something that would be a low viscosity liquid that would inject very easily and then gel in situ. This system demonstrated some glucose-dependent release of insulin, and also demonstrated the ability to do some correction following injection and glucose tolerance testing. And so this was kind of a way to improve on another one of these undesirable features of that original insulin system by adding an ease of injectability that would maybe make it more easily used along with a pen injector, standard syringe injection, et cetera. Professor Gu also mentioned the use of microneedles. We've actually taken some of these network polymers and used them to fabricate microneedles as well. We can make needles without the need for covalent bonding, just using PBA diol interactions to cross-link the network. Again, this would be another strategy to maybe make a more ease of use centered approach to insulin delivery, where you could have a wearable microneedle array that could swell and release insulin as needed. In order to provide insulin, in response to glucose levels and things like that. And so this was kind of just one proof of concept demonstration of that approach to try to improve on that. The last thing I'll talk about is actually unpublished data that we're very excited about, some new stuff that we've been doing. And this is in the area of making smart insulin polymer formulation, right? And so we can think about polymers and insulin and mixing them. And again, this desire to have insulins that we don't have to do a lot of really low yielding modification chemistry on. Can we actually think about, high yielding insulins and this kind of stuff? And so we want to think about mixing insulin with polymers and doing so in a way that could produce complex, right, or material. And so we've screened some different insulins. I think we have insulin 1, 2, and 3 here. We're able to find an insulin where we mix it with the polymer at, roughly equivalent ratios, 1-to-1, and we can get the formation of complexes that make these kind of insoluble aggregates. We can image them by TEM. And these seem to be basically kind of net neutral pH. So when we mix them together, and the complex has a sort of an isoelectric point right around neutral pH, which should kind of facilitate in the body a stable complex between the insulin and the polymer. These complexes, thankfully, are also glucose responsive. And so as we increase glucose, we modulate the interaction between the insulin and the polymer, and we can actually make them soluble. And so we can see things like, the complexes don't really form in the presence of higher levels of glucose, or if we take the formed complexes and we put them into an elevated level of glucose, we get glucose-dependent release of insulin from those formulations. We can do things like tuning the alternating between low or no and high glucose concentrations, and we can see differential response in the release rate. And the insulin that's released looks like recombinant human insulin, right? It has that sort of characteristic alpha helical secondary structure as well. And so we take these smart insulin polymer complexes, and in this case, we'll mix them at 1:1 insulin to polymer and 1.5:1 insulin to polymer, just kind of picking two ratios that were pretty close to equivalent. And we take and we treat these mice at time 0, and we then do a series of glucose tolerance tests, and we can track things like area under the curve, and we can see, over the course of a day, we're able to continually correct blood glucose following a single administration at time 0. Both insulins, both kind of formulation ratios look kind of comparable in this data. We can then start to look at things even a little bit longer out, right? And so we can compare this to kind of one of the clinically used kind of the longish acting insulins, insulin detamir, dosing everything at time 0. Initially, these are very fasted mice. They're fasted about 12 hours, I think, before treatment starts. And so blood glucose levels go down. We picked a dose of detamir that would get to roughly the same level of blood glucose at 3 hours, sort of a similar potency. If you think about how they would typically measure kind of an IU potency scale, we picked a dose of detamir that would get us kind of comparable levels of blood glucose at 3 hours. We then do glucose tolerance test at day 0, demonstrate that the detamir, there's a lot of detamir on board. The detamir is still able to kind of correct. It looks pretty similar. We do detamir dosing every day. And then we go back at day 2 and we do another glucose tolerance test. Our insulins are still responding. We go back at day 4 and we do a glucose tolerance test. And our 1:1 insulin formulation actually is still looking pretty good. It's got very tight area under the curve. It's not really changing much over the course of 4-ish days since treatment, right? And so this looks pretty good. One thing we wanted to show was, is this really a glucose responsive insulin effect, or do we just make insulin with a really long tail of activity? And so we go at day 0, day 2, and day 4, and we measure serum insulin levels. And you see here at 30 minutes before and then 30 minutes following a glucose tolerance test, serum insulin levels are going up at all of the days measured at day 0, day 2, and day 4. We're seeing an increase in serum insulin levels following these glucose tolerance tests. So we want to kind of zero in on day 2, sort of that intermediate time when we wouldn't expect a lot of residual insulin on board from an immediate injection. And we wouldn't expect sort of a long tail kind of effect either. We kind of have something in the middle. So if we take the data and we treat these mice after 48 hours, we administer a glucose tolerance test and we do a kinetic profile of serum insulin levels in the mice. And we kind of track that with blood glucose level. What we see is about a 3x enhancement in the serum insulin level following blood glucose challenge. This is at 48 hours after the insulin has been administered. If we compare this, just to make sure there's not any residual mouse insulin going on, we've got the dotted lines are for just sort of an STZ mouse that hasn't been given any insulin at all. And so we do see a glucose-dependent increase in serum insulin levels. I should point out that these data points are from different cohorts of mice, and so kind of the sum of the jump up and down is just a little variability in the whatever cohort was chosen for the different time point. It turns out mice don't have enough blood to bleed at every single time point here and not kill them. And so we had to pick, these are different cohorts. And so the small variations up and down are kind of normal and expected. We can then take and look at this insulin in the course of redosing every 5 days. What we can see is we're able to maintain the mice in a state of normal glycemia for over a month with dosing every 5 days. Whereas the controls and the treatments of daily detamir and these kinds of things aren't able to do the same kind of correction. Even though this is a potency adjusted dose of detamir, the one that would still get us in a fasted animal was at the same level as the insulins we're using. Some very exciting and very preliminary data we've recently got is actually taking these insulins and putting them into these insulin formulations and putting them into pigs. We can do things like an oral glucose tolerance test, and at pre-treatment, these pigs show a trace in the black here. And then we treat at day 0 and we monitor the response to an oral glucose tolerance test for a week, right? And so we have data at 0, 1, 2, 3, 4, 5, and 6 days. So for a full week of coverage, we're able to actually have much tighter control following an oral glucose tolerance test in the pigs than we would have in a pre-treated diabetic state. These are Aloxan pigs, so they've been induced to be diabetic. And just to kind of zoom in on two of the pigs, this is 3145 and 3112. You can see we have their data, their 2 days pre-treatment and their pre-treatment data there in black and gray. And then you can look at their blood glucose levels. These are the raw blood glucose levels in the days following treatment and see how they respond to oral glucose tolerance tests for that week after they've been treated with our insulin. And they are much tighter in control. Their levels are quite a bit more normal. These Alborz swine have a certain extent of insulin resistance. And so I'm told that 3112, because it has a higher set point, it's probably a little bit more insulin resistant than 3145, based on the fact that it started around 300 versus the 3145 was around 200. But we're kind of seeing examples here of the ability to control blood glucose in these swine out for a full week after a single injection. And so I guess where are we going here? Where are the drug delivery scientists going? I would say we have a few things we have to really improve. Right? We have to improve if we're going to use materials for delivering insulin. We have to improve insulin's availability and release as a function of blood glucose level. Oftentimes our release data is shown for many hours, and we may want our insulin faster than that. And oftentimes our systems leak, right? And we don't want insulin to leak when we don't need it. This is potentially a problem. If I'm a materials scientist, and so we're always going to use polymers or materials or hydrogels or these kinds of things to deliver insulin. We may have to think about, the compatibility of the materials we're using. If I'm going to make my materials on the basis of protein type interactions, things like kinase or lectins, these kinds of things, glucose oxidase, I have to be worried about immune responses. Maybe I have to be really conscious of, I talked about injection pressures and things like that. We have to make sure our things can be used. And, we might have to contend with somebody using this for a lifetime, right? So, putting this into a juvenile individual and having them use our insulin for 30, 40, 50 years, right? They're going to have to use it and not have any complications that arise. And then I think moving towards bio-inspired insulin therapeutics, thinking about co-delivery with synergistic hormones, things like amylin analogs, which I talked about very briefly. Liver-directed insulin function. I know the first talk today talked a little bit about some of the liver specific requirements of insulin. And then thinking about sort of integrating this abiologic and biologic communication using, a little bit more smart therapies so that we don't have to put as much onto an individual to have to measure their blood glucose, do these routine monitorings, et cetera. And so I would say this is still a challenge, right? People have been working in this area. Drug delivery scientists have been working in this area for over 30 years. And we haven't sorted out a great way yet to deliver insulin. I think it's still not perfect. And so we're going to keep working on it. I think we're going to keep synthesizing new materials and new strategies for delivering insulin to try to address all of these points. And this has been a big challenge. And then, of course, another arm of my group that I won't talk about today thinks a lot about glucagon. And this really remains an area that's almost completely unexplored. I know Zhen Gu talked a little bit about a dual hormone patch, but maybe there's a way to integrate this as well as sort of a yin and yang kind of approach to managing blood glucose in a more biomimetic way as well. And so with that, I'll end where I started. Thank the group, thank funding, thank JDRF specifically for support and really championing this work over the years. And hopefully I left time for questions, although I have no idea how long I took.
Thank you very much indeed. Very fascinating talk. We actually had one question in the chat from Andy Chapman from Carbometrics. So he wondered if you have tested beta-hydroxybutyrate with any of these boronic acids. Of course, diabetic ketoacidosis might be a challenge. And perhaps I let you answer that first. He also asked about glycosylated proteins.
Yes, the answer is no to both. Unfortunately, we haven't looked too much at glycosylated proteins. We haven't looked at beta hydroxybutyrate. We were kind of looking at primary analytes, but I guess this is a good, this point about elevated ketose levels of ketosis in diabetics is certainly well taken. We wouldn't want to have chemistry that where some undesirable metabolite was also triggering accelerated insulin function either, I think. And so, certainly something to think about for sure. Glycosylated proteins, no. I do have a little bit of concern though, because some of the chemistry we do to open the glucose ring onto the amines of the macromers, which is the chemistry we're using, kind of that gluconic delta lactone derived diol for cross-linking, is a similar mechanism to how a lot of proteins get glycosylated anyway. And so it is possible that some of these phenylbronic acid are also interacting with glucosylated proteins, glycoconjugates on cell surfaces, et cetera. So.
Thank you. Well, one perhaps naive question from someone who is not a chemist. So you talked so much about the glucose sensing part of a smart insulin, and what I understood is how difficult it is to get PBA to really do the job. So is this the major challenge of the smart insulin, or is it equally challenging to get the insulin release component right?
Yeah, I mean, as the drug delivery scientist, I think I would focus more on the release component for me. I think glucose specificity and sensing is also a big deal. I'm certainly very excited about the lectin molecules that were mentioned in the first talk, and then also in the previous talk by Danny. I think those are going to help address some of these glucose sensing and glucose specificity challenges. I'm really hopeful for some of those macrocycles. But as a drug delivery scientist, if we're going to think about putting stuff into a polymer, diabetes and insulin release is one of the most challenging kind of kinetic situations to be in, where you have, this temporal importance to have release exactly when you need it, right? And so I sometimes use the example of like controlled release of a chemotherapy or of a cancer, inhibitor or something like that, where I don't need to have it at the exact moment when I need it, right? I just kind of need to be able to control it within a therapeutic window, right? The concentration systemically. For insulin, it needs to happen exactly, and not only at the right dose, but the exact right time in order to really correspond with changes in blood glucose. And so I think that makes it quite a bit harder than what we typically do in the drug delivery field. And so I think that's why this is a fascinating problem. I also think it's why it's a very, very challenging problem, where people have been trying this for since Kataoka and before, in the early late 80s, early 90s, using PBA-based systems for insulin delivery. And here we are in 2023, and I'm giving a talk on PBA-based systems for insulin delivery. I think we still have some challenges. So.
Okay, thanks very much. I think we stop it here for now. We will still have a discussion at the end of the symposium, and then could also talk about, quick insulin release is a challenge, but then also preventing hypoglycemia. So stopping insulin action might also be a challenge, and I think that has been partly addressed, but we could discuss it further. So for now, I think we have a break. We are slightly behind time, so I think we extend the break to 15 minutes. So we convene at 25 after the hour, and then we'll have the next presentation. And I look very much forward. Thank you to all the speakers so far, and I look forward to another bunch of great presentations after the break. So see you here at 3:25 p.m. UK time. Thank you.
Welcome back, everybody. It's now 3:25, and I now want to ask John Buse, who's another one of our scientific expert who's very kindly supporting this work, to introduce Bob Langer, who I think is giving a pre-recorded talk. Many thanks for joining us, John, and I'll ask you to introduce the next talk.
Thank you. So, I often say at presentations that it's my honor to introduce the speaker. This time I really mean it. Bob Langer, as you heard, is a mentor to several of the authorities that have presented to us today, but really is almost at that sort of household word kind of level with regards to his impact on society. He's one of 9 institute professors at the Massachusetts Institute of Technology, which is the highest honor that can be awarded to a faculty member at that institution. He's published over 1,500 manuscripts, which have been cited nearly 400,000 times, creating an H-index of 309, which is apparently the second highest in the world, highest among engineers. He has almost innumerable patents, but over 400 companies have licensed his patents. And, I would tell you about his awards, but there are over 220 of them. He holds 41 honorary degrees and is a member of the National Academy of Medicine, the National Academy of Engineering, the National Academy of Sciences, and the National Academy of Inventors. I don't think there are enough superlatives to describe how well qualified Bob is to present to us today. And though he wasn't available to be with us, he was gracious enough to record the following presentation. Massimo, can you run the presentation now?
Thank you so much. It's a pleasure to be able to speak with you today. I'd like to tell you about some of the work we're doing in the diabetes area. If I could have the first slide. Next slide, please. So I've been involved in doing research that would have effects on diabetes for almost 50 years. The very first paper, one of the very first papers I wrote with Judah Folkman was isolation of the very first angiogenesis inhibitors. And that would many, many years later, because of the work at Genentech and other places, would lead to really the treatments for diabetic retinopathy. Also, that same year, we published an article on the very first delivery systems for macromolecules, which would lead to diabetes treatments for GLP-1 agonists. And so that's also been important. Separately, it also led to, delivery systems for COVID vaccines and really all messenger RNA technologies. But these were some of the early things that I got involved in. Now, more recently, I'd like to highlight on two areas that I've worked in for the rest of the talk. Next slide. One of these is, of course, children and adults. You can't take a molecule like insulin orally. The reason for that is that, and you're seeing this in this sort of diagram and video here, is that whenever somebody takes a large molecule like insulin, what happens is that it first, proteases will degrade it. Secondly, it's so big that it really can't get through the intestinal tract. So that's been a problem. But Gio Traverso and myself had an idea that we thought might change that. And really the idea is based on the fact, if we could go to the next slide, that people inject drugs into the stomach, into the gut all the time. He's a gastroenterologist, and he does that himself. Some of the advantages of injecting into the gut are there's very high delivery efficiency, no painful sensations, and rapid tissue regeneration. The only problem is whenever somebody does this, you probably go in with an endoscope and you go in through your rear end. Well, we want to go in through your mouth. How might you accomplish that? Well, let me show you an idea we had on the next slide. What if we made a little pill? And I actually have one such pill here. And this would be a little tiny pill but inside this pill, you see the green, which would be like a needle. It's a needle made of insulin in this case. And behind it, you see this post, which just attaches it to, and it's biodegradable. It just attaches it to a spring. And the spring is, you can see, is in the orange area. And the idea is that the spring, if it's cocked, it would just shoot the needle right out. But you don't want it shot out right in the beginning, because when you swallow it, it's in your mouth. What you want to do is maybe have it shot out, an hour later when it's in the stomach, to achieve what we saw on the last slide. So, and the idea is the spring is so big, it can't get out of that little hole where you see the 5 millimeters at the bottom, but the insulin tip can. So that's what we did. But you might ask one other thing. How do we know that when you swallow this, that the part where you see the 5 millimeters will always face the stomach wall, so it can shoot this out rather than otherwise. So what we did is we borrowed from nature. If you have a leopard tortoise, if we could go to the next slide, what happens is it will, because of its weight distribution, it will always self-orient because certain things are going to be more unstable, so it will turn over and do just what you see here. So the idea is you localize it, you inject it, and then it dissolves. So it's self-oriented. Just so you believe me that it's not just a picture. You know, we publish this in Science, but I'm going to actually do a demonstration where I take this, and no matter what I do, I don't know if people can see this, but no matter what I do, it will always fall that way over and over again. We've done this thousands of times, and it always falls that way just because of this self-orientation concept. Next slide. And that's just a picture of what the leopard tortoise does. And the idea is that sugar actuator, if we could go to the next slide, the idea is that what breaks the sugar, it's a glass that's holding the spring and prevents it from not shooting out. But then you can break it, you know, by osmosis. Water goes in over an hour, breaks the sugar glass, and then you see this is the spring. And like I say, it can't get out of our little system because it's too big, but it sure can shoot that needle out. So that's what we did. And just to show you some data, next slide. Here, and this is all published in the journal Science, and we've actually published articles now in different Nature journals on delivering other large molecules, but I want to highlight insulin here. But what you can see is that in pigs, that's the animal model we used when we did many of them, you can get the same plasma levels of insulin as you get with a regular injection through the skin with exactly the same amount. And so we call this SOMA. It's a self-oriented medical applicator, and you can see that that you get the same amount by doing this as you do SC, which is subcutaneous. You also see on the right, the histology is perfectly normal. It doesn't cause harm. Next slide. And not only do you get the same, next slide. Sorry, go back one, please. So not only do you get the same insulin levels, you also get the same, statistically at least, blood glucose levels. And here you see the blood glucose changes when you compare subcutaneous to the oral system. Again, you see no statistical difference between the SC and the SOMA, or intragastric as well. That's IG. And again, when you look at the histology, it's perfectly normal. So this is what we did at MIT. We have a clinical partner, Novo Nordisk, which has helped fund the work and really has done a great job developing it. And they have now begun to look at the safety of this in humans. And they've now done 65 patients. With so far as we can see, no ill effects. So we're very excited about hopefully someday perhaps having with this or some variation of it, an oral delivery system for insulin. Of course, another problem, and the Juvenile Diabetes Foundation came to approach me a number of years ago, is could you ever create an artificial pancreas? Well, people have tried before. Next slide. And one of the things they did is encapsulate cells in what's called beta cells in a semi-permeable membrane. And you can see the idea here that insulin, which is, say 6,000, and glucose, which is much smaller, can diffuse back and forth in such a membrane. And the cell is immunoprotected. The transplanted cell on the top is immunoprotected because when you look at those pores that the glucose and insulin can get in, antibodies are too big to get through them. So are immune and inflammatory cells. So that's the idea, that you could make such a device. One of the challenges, though, is what material should you use? One of the very popular ones, next slide, has been alginate. Alginate is shown here. It's in seaweed. It's been used in lots of things, but the beauty of it is when somebody does encapsulation, almost always you use organic solvents to encapsulate things. But the beauty with alginate is you can make a capsule by putting it in water. And then if you inject it into a bath of a divalent ion like calcium or barium, it will form a beautiful bead by what's called ionic cross-linking. And the next slide just shows that. You can take an electrostatic droplet generator, put cells in the aqueous system, and that's really important that it's an aqueous system, because organic solvents will damage cells. But when you do that, it shoots it into this bath, and look at the beautiful beads with the cells encapsulated on the right-hand side. So that's been exciting, but there's been one problem, and that is that the problem has been that alginate, and many companies have started based on it, but alginate causes a reaction, a tissue reaction that you don't want to have, and blocks off the insulin from getting through because of these fibrous responses. So that's not so good. But the Juvenile Diabetes Foundation approached us, and I asked Dan Anderson, who was then one of my postdocs, he's now a full professor, we had some ideas on maybe we could make lots of alginate derivatives, literally thousands, to take advantage of the idea that you could still do the water-based encapsulation, but you could now maybe come up with some that would be more biocompatible. Now, whenever you do something like this, like high throughput, you need to do two things. One is you need a rapid way to test it. And so what we did, next slide, is develop what we called a high throughput mouse. Actually, it's more like a medium throughput mouse, but the idea is you would inject our alginate derivatives, and if there's going to be free radicals that would react to O2, or cathepsins, which are indicative of an inflammatory response, and then you put in certain dyes like luminol, well, the areas where you put the alginate will light up, and you see that here. But if they don't light up, that's at least an indicator that it's biocompatible. So in other words, this would be a medium, a pretty good test system. You could do 1,000. If you'd had 10 of these per animal, you could do 100 animals and test 1,000 new alginates. Next slide. So that's part one, is that you have to have a pretty rapid way to test it. Of course, the other thing is have to have a pretty rapid way of making all these alginate delivery systems. And we did that by actually having robots do the work. They work 24 hours, even though our students are pretty good, they don't work as much as that. And we made, we really made doing this many different alginate derivatives. Just to show you a few examples on the next slide. So you can probably make 1,000 of these, and they all have this ability to do ionic cross-linking, but some, our hope was, you'll get better biocompatibility. And so I'm going to just show you a flow sheet that we made to illustrate what happens and the whole flow of what we do on the next slide. So what you do is you start out with the basic alginate, but then you derivatize it, which will change what the cells see on the surface by amidations, esterifications, and click reactions. And this was all done even before the Nobel Prize was awarded for these click reactions last year. Of course, the people that won it, especially, that did the pioneering work on the click reactions. They deserve the credit for developing it. We just used them, but they were very helpful. And then we have this robotic system that I showed you next to it. We characterize the materials many ways, and next, then we make the capsules like I showed you next. Now, if you go to the bottom right of the screen, we have that high throughput or medium throughput mouse where we can look at immunoresponses. Then we take the best performers of those and put them in the peritoneal space to see if they still are compatible. Then we actually put islets in them and test them in mice, and then finally test them in non-human primates in large animal studies. Next slide. And what you can see is if we use regular alginate pre-transplantation, you get very nice capsules. But on the right, what you see is basically the cells die. You get fibrotic overgrowth and the cells just die within 4 weeks. But if you take the MIT system, next slide, what happens is it's much more. Notice that now you retrieve them, and the capsules are really clean. There's really no fibrosis 4 weeks after retrieval. This is the best ones out of maybe 1,000 that we did. Almost all of the ones we tested didn't work, but we found 3 that actually work quite well. Next slide. And if you look at viability, what you see here is all but one have the same viability post-transplant as pre-transplant. Next slide. And if you do this after to look at intraperitoneal effects in non-human primates, you see very few macrophages in myofibroblasts, and even after intraperitoneal administration, 6 months later. Next slide. And this was a study we did with Doug Melton at Harvard, showing that you can actually treat with our systems, you can actually treat diabetes and really, in essence, cure it by doing this. Here you have blood sugar levels for over 150 days from one implant. Just to really graphically illustrate how these look, we also put them on FDA-approved catheters and coated them with these polymers. Next slide. And you see that in the controls, you get a lot of fibrous overgrowth, but in our catheters that we treated with the best systems, you get none. And on the final slide, if this is for the chemists in the audience, these are the 3 derivatives that worked. And like I say, there's probably close to 1,000 that didn't. We've actually published a lot of this work, at least, in 9 different Nature journal papers, going over everything from the right shape, the right animal models, different coatings, different alginate derivatives that do work and don't work, and many other things. And there's even a company now, Sigilon, which we hope is working with Eli Lilly, to hopefully bring this into the clinic in the near future. Thank you so much. It's a pleasure and an honor to be able to speak.
So that was outstanding. Clearly, we're not going to be able to have Dr. Langer answer questions, but do hold your questions until the end, and certainly the discussants in general would be happy to address them to the best of their abilities.
Great.
Simon, back to you.
Thank you, John. And yeah, I agree. That was a really fantastic talk. And hopefully some people might talk to it at the end. So we're now going to move on. And Reinhard, you very kindly agreed to chair one of us, another speaker. So if you could introduce Michael Weiss, I'd be grateful. Thanks.
Dear colleagues, on behalf of the advisory subgroup, once again, I have the privilege to welcome a distinguished speaker to our symposium, Professor Michael Weiss. Prior to his appointment at Indiana University, Dr. Weiss was chair of biochemistry and the Corn Bloom Professor of Cancer Research at the Case Western Reserve University School of Medicine. As of today at the Indiana University, with his background in human medicine, physics, and administration business, he fills the dual role of chair of the Department of Biochemistry and Molecular Biology, and also director of chemical biology and biotherapeutics for the Indiana University Precision Health Initiative. But Michael, I understand you're also acting as entrepreneur, and so in 2009, you founded Tamerlane Diabetes, which designs and develops novel insulin analogs, and you're currently also serving as its Chief Innovation Officer. I assume in this latter faculty, you're going to report on your endeavor on ultra-stable insulin analogs and their clinical applications. And so I may say, welcome, Michael, and thank you for joining us. The screen is yours for the upcoming 20 minutes. Mike, please.
Thank you very much. It's a great privilege to be here, and really exciting to see all the wonderful young people that are in this field and their enormous promising technologies. And I want to thank Danny for his mention of our efforts in glucose responsive insulins. However, today I'd like to focus on some basic chemistry and structural biology of insulin stability as a enabling technology for diverse clinical applications. And I'd like to share my screen. I have one disclosure, Thermalin Incorporated, which I'll mention at the very end of the talk. I'd also want to express my gratitude to the JDRF for its Career Development Award 33 years ago, which enabled my junior faculty career to begin, and especially the enormous inspiration provided by the 1990 Monaco meeting by JDRF. I'd like to show you a picture of our team at the Indiana University School of Medicine, shown here in front of our Chihuly sculpture of DNA, including our collaborating laboratories that I'll mention at the end. And I'd like to thank the NIH, the Helmsley Charitable Trust, and the Lilly Foundation Insight Program for its support. Now, I was very lucky to spend a year as a graduate student in the Department of Biochemistry at Oxford University, where I got to know people in the Hodgkin's group. And for the rest of my career, Tom Blundell, Eleanor Dodson, and the late Guy Dodson were part of the family of mentors in the field of insulin. And I think that the field, the culture of the field, it has an enormous debt to Dorothy Hodgkin for her inspiration. The crystal structure of insulin was the third crystal structure of a protein to be determined in the late 1960s. The high resolution structure in 1971 by Blundell et al. Is shown on the right. And I think it was the first really beautiful protein crystal structure due to its striking six-fold symmetry. This is the T6 insulin hexamer with two zinc ions aligned in the middle. And Tom Blundell, who had studied architecture before going to graduate school in biochemistry, likened its elegance to the rose window at Notre Dame. And with thanks to Pierre de Metz, you can see the resemblance. Guy Dodson himself was had a spirit of a poet, and in a caption in a 1972 review with the Hodgkin group, likened this self-assembly process to the first line of Keats's romantic poem, Endymion, "A thing of beauty is a joy forever." And our goal today is to take advantage of this structural beauty to investigate the stability of insulin and to explore unnatural ways with an extended genetic code technology to enhance the intrinsic stability of the insulin monomer. To do that, we need to look in part at what are the mechanisms of degradation. And here the field owes an enormous debt to Jan Zbrangi and his pioneering work at Novo Nordisk. So in the 40-some years since my graduate work, the world of global health has experienced the challenge of a global diabetes pandemic. And this has put a spotlight on challenges to the global distribution and use of insulin, excuse me, which is defined by the WHO as one of the essential medicines for global health. And the WHO predicts an enormous increase in the need for insulin, primarily in the developing world, but also in North America and in Europe. In much of the developing world, insulin is stored in clay pots because the majority of villages lack access to electricity and refrigeration. And online at the Lancet Diabetes and Endocrinology is a commentary on provocative new studies from the Atkinson Group and colleagues on the real world insulin stability and its implications for global access. But I'd like to mention that the instability of insulin is not only an issue for distribution, but also a limiting factor for innovative insulin delivery technologies. So I'd like to talk about three things: single chain insulin analogs, unnatural mutagenesis, and translational implications. So the evolution and divergence of insulin sequences has been constrained by multiple factors, including function, binding to the receptor, foldability of the proinsulin precursor in beta cells, assembly in the secretory granules, and the hidden propensity of the molecule to toxic misfolding. So given these constraints, how could we improve on the native structure of insulin? And our first approach is to use foreshortened C domains to make the molecule topologically incompatible with cross-beta assembly, the key step in the formation of an insulin fibril. Insulin fibrillation is the major challenge above room temperature. And we have been exploring for many years the structure and function of single-chain insulins as an ultra-stable form of insulin compatible with intraperitoneal long-term pumps. And here is an image of an insulin molecule tethered by a 6-residue foreshortened connecting domain whose sequence has been optimized to reject binding to the mitogenic IGF receptor. And the essence of the single-chain insulin robustness lies in the biophysics of protein folding and misfolding. We now know that in addition to the native folding funnel shown here on the left, there's a coupled landscape of aggregation mediated misfolding. And in fact, the amyloid fibrils may represent the universal thermodynamic ground state for polypeptides as a class of heteropolymers. And so the question is, how could we keep insulin molecules at or above room temperature from forming fibrils that could clog the catheters and lead to pro-inflammatory precipitates, for example, in an intraperitoneal pump? And so this is the region of the conformational landscape that we wanted to explore with biophysical studies. And in these back-to-back JVC papers, we demonstrated that ultra-stable single-chain insulin analogs may be compatible with native signaling duration and reduced mitogenicity, and be incompatible with formation of an amyloidogenic nucleus, unable to form microfilaments or mature fibrils, and yet still have sufficient conformational play to assume the active conformation on receptor binding. We began collaborating with Rob Tico at the NIH, who has pioneered the development of solid-state NMR techniques to interrogate the three-dimensional structure of protein amyloid, as exemplified here in his studies of beta amyloid peptide. And we undertook a systematic series of isotope-directed solid-state NMR studies to map out regions of beta sheet and non-beta sheet in the A chains and the B chains of insulin fibrils. Here's an example of a chemical shift mapping approach in which we compare and contrast the native chemical shifts in solution to the beta sheet characteristic chemical shifts of insulin fibrils. In collaboration with Steve Kent, then at the University of Chicago, now retired, we made a series of samples, each containing a single C13 carbonyl isotopic label. And the reason for this is to apply a solid-state NMR diagnostic technique for looking at parallel in-register beta sheet in amyloid. This experiment gives a positive outcome if and only if you have parallel in register structures, because then the myriad C13 labels would be aligned at close proximity. And here are data in the B chain for single carbonyl labels at B6, B11, B18, B23, B24, B25. And in each case, the distance implied by the relaxation properties of these C13 atoms can only be accommodated by a parallel in register model. And the same conclusion was reached in two labels in the A chain, one at A3 in the N-terminal helix in the native state, and the other at A16 in the C-terminal helix in the native state. And so what this implies is that there's a major conformational conversion ongoing from native insulin with its anti-parallel C-terminal B-chain beta sheet to the parallel cross beta sheet register of the mature amyloid, which extends to both the B chain and the A chain. So how can one prevent such cross beta assembly? Well, it turns out that any of a set of models consistent with cross beta assembly that's parallel and in register requires the distance between A1 and B30 to be greater than 30 angstroms. And so therefore, any foreshortened C domain less than 30 angstroms would be incompatible with this mode of assembly. And so we picked 6 residues, here shown EEGPRR, in order to provide enough play between the A chain and the B chain to enable site 1 occupancy in the hormone receptor complex, which is the key trigger for the choreography of conformational change in the receptor that ultimately leads to transphosphorylation of the intracellular cytoplasmic tyrosine kinase domains. Now, the proof is in the pudding. This is a in vivo-based thermal stability assay in which single-chain insulins or control analogs are mildly agitated for days, weeks, or years at 37 degrees centigrade and then tested for activity in diabetic rats. And this was in collaboration with my Case Western Reserve colleagues, Professors Behrmars Ishmael Beghi and Nelson Phillips, shown at bottom right. And here's one example of a basal insulin SCI that is compared to Lantus. And whereas 11 days of gentle agitation at 37 degrees led to the complete inactivation and fibrillation of Lantus, the ultra-stable basal SCI was fully active after 3 months of heat stress. And we've actually extended these experiments for more than a year. Now, a single chain molecule is reminiscent of IGF-1 or IGF-2, and indeed their modes of receptor binding are analogous. The foreshortened C domain tether actually gets displaced from a groove between the A and B chains to enable insertion of the alpha-CT element of the ectodomain alpha subunit, recapitulating the mode of native insulin binding. And here's a microcrystal receptor structure recently published in the Biophysical Journal that verified the molecular dynamics simulations that we had published in 2018. And here are some details of how this threading of alpha-CT through the loop formed by displacement of the C domain occurs structurally. So I'd like to thank our team that includes key members in Australia, John Menting and Professor Michael Lawrence, Professor Brian Smith and Dr. Nick Smith, and at Indiana University School of Medicine, Bala Dalyan and Mark Jarasinski. So conclusions from this part of the talk: insulin fibrils exist parallel in register beta sheets. Topology of single-chain insulin analogs resist fibrillation even at elevated temperatures. As an aside, we now have such analogs that you can boil for 20 minutes without loss of activity. There's a sweet spot for C domain length to enable receptor binding and yet prevent fibrillation. And the SCIs can be made to be rapid-acting, basal, or even biphasic. I'd like to switch gears to unnatural mutagenesis, going beyond the 20 standard amino acids, and in fact, focusing on the so-called 21st encoded amino acid, selenocysteine, which is natively used in the thyroid gland in the course of thyroid hormone biosynthesis. And this work was done together with Professor Norman Matanis from Hebrew University in Israel and his student Orit Weil-Katorza. And what we discovered is that replacement of cysteine A6A11, and only that cysteine, by a diselenide bridge stabilizes the insulin monomer and delays fibrillation. And the stabilization is both thermodynamic, probed here by guanidine denaturation studies at the top, but the receptor binding surface is not perturbed. And so the affinity for the insulin receptor, and indeed the potency in streptozotocin rats, is indistinguishable from native human insulin. This is not true with replacement of external cysteine B7A7, which exhibits no increase in stability. Now, one can apply this idea generally to all clinical insulin analogs in current therapeutic use. And so here's an example of putting that A6A11 diselenide bridge into Lispro, glulisine, and aspartate. And in each case, there is a 1 kcal or more of thermodynamic stabilization, and also a delay in the lag time before fibrillation at 37 degrees C. So why does this happen? Our hypothesis is that there are subtle packing defects around the native A6A11 disulfide bridge, as first noted by Dorothy Hodgkin and colleagues in their classical crystallographic studies, and the slightly larger atomic radius of the selenium atom and the slightly longer bridge bond length between two selenium atoms relative to two sulfur atoms leads to more efficiency of core packing. And this hypothesis would also rationalize why a diselenide bridge on the external surface of insulin, i.e. B7A7, has no effect. Now, evidence for this enhanced core efficiency is provided by NMR. So the NMR spectrum of insulin monomers are remarkable for a large variation of amide resonance line widths, reflecting millisecond motions in the core of the protein. And you can see these broad amide resonances at top left. And when one puts in the diselenide bridge, they become sharper and more uniform. And in fact, the diselenide bridge slows down amide proton exchange in D2O solution, both near and far from the modified bridge. And here our stack plot of 1D NMR spectra showing progressive replacement of NH by ND in D2O solution, which is slowed down broadly in the molecule by the diselenide bridge. We interpret this as that the slightly larger bridge damps conformational fluctuations in the protein core. And here's some data. In green is the diselenide analog, and in black is the native analog. These data were obtained using Insulin Lispro, the active component of Humalog, as our test system. So conclusions here: selenium A6A11 is stabilizing relative to a disulfide bridge. Such stabilization is site-specific. The unnatural bridge damps conformational fluctuations, presumably related to enhanced packing efficiency, and this effect generalizes to all clinical insulin analogs in current use. Translational implications. So we're just beginning the second century of innovation of insulin analogs, as so beautifully shown in the previous talks in this symposium. And enhanced intrinsic stability of the monomer itself can enable further improvements in insulin formulations and delivery, in particular by enabling zinc-free formulations of monomers and dimers in millisecond exchange. And this also would confer flexibility in the choice of excipients, including active excipients, to promote ultra-rapid absorption in the bloodstream. Now note that if we don't need zinc ions, and we can make the insulin monomer less sticky and prone to self-association, then in principle, we can have ultra-concentrated, ultra-rapid insulin formulations to miniaturize pumps. And so here is an example of an ultra-stable monomer in a zinc-free formulation with an active set of excipients at U500, which is faster on and faster off than FIASP, which we picked as a gold standard for a reformulation of a rapid-acting analog designed for use in closed-loop systems to enhance the safety and efficacy of the control algorithms. So conclusions here: stabilized insulin monomers enable zinc-free formulations with extended shelf life. Such formulations broaden range of compatible excipients. This approach enables development of ultra-rapid U500 therapeutics, and these in turn may permit miniaturization of insulin pumps. So I'd like to acknowledge my colleagues and coworkers at Indiana University. And I mentioned our collaborators at Hebrew University. And I'd like to make a special thanks to my colleague in chemistry at Bloomington, Richard DeMarchi, and his former postdoc, Alex Sakoff at Novo Nordisk, for their invention of the DES-dye synthetic route to 2-chain insulin analogs, which has enormously enhanced our throughput in our synthetic studies. So I'd like to end as I began with a picture of our IU team. And I think the excitement of today's symposium and the promise of these new technologies is what make us so enthusiastic day in and day out to contribute to this second century of insulin innovation. Thank you again for your invitation.
Thank you very much, Professor Weiss. This is Reinhard Becker speaking. So that was an excellent talk, and you introduced us to a problem which we were not aware of, and you already provided the solution of these very stable insulin analogs. Now, having worked in the pharmaceutical industry for quite a while, I remember that in the very beginning there was a Lispro, and the Lispro was developed with zinc-free formulation. Unfortunately, it wasn't stable enough, so zinc had to be added, which therefore lost a little bit of its accelerated absorption, but it was still enough to be faster than the regular human insulin one. And in our company, which is Hext and then Sanofi later on, we had developed a rapid-acting product completely without zinc. But of course, it was very sensitive. Adding zinc, it lost the rapidity of absorption, and was slightly like regular human insulin if you added that zinc. So I think it's fascinating because you are producing a really stable insulin which eases manufacture, at least the shelf life of products, which I assume. And I guess you have already tested some of your products on the shelf and compared it to the conventional insulin products.
Yes. And I actually appreciate very much the elegance of glue lysine and the properties of Apidra as a zinc-free insulin formulation, which was pioneering in that direction. So we are very interested in a new kind of fibrillation protocol. So previous studies by us and by others would aggressively put an insulin at, say, 37 or 45 or 50 degrees continuously for minutes to hours in order to do lots of experiments per week. But that's not what really happens in the real world. In the real world, an insulin vial may be at room temperature or 30 degrees 99% of the time, and then have transient exposure to fluctuations, for example, in the car on a hot day driving home. And so our new protocol, which shows much greater discrimination among analogs, is to mimic this transient thermal challenge and then go back to 30 degrees. And the idea is, with this challenge, for say 20 minutes at 45 degrees centigrade, one might make seeds. And these seeds would be invisible to standard analytical techniques, except by measuring the lag time when you go back to a lower temperature. And this sort of real world inspired protocol is what's uncovering subtle differences among analogs and among excipients.
It's very interesting. Thank you very much for this comment. I've seen, there is no question from the team, so this leaves me to think again.
Reinhard,
yes,
sorry to interrupt you. There's one that's just come in from Simon.
Just coming in. Let me see where we are here. The question is concerning clinical studies to make a rapid-acting insulin analog truly a rapid one. Well, this question, of course, in my view, has two sides. In my view, always the Lispro was the fastest one as a true monomeric one. And to accelerate this, well, you need to do different things on the excipients, but certainly your opinion on this is being asked.
So I don't believe that there are any scientific barriers now. There are only regulatory barriers and barriers to entry because of the greater complexity of a new molecule rather than a new excipient. However, you know, at the NIH meeting on closed loop systems last week that one of our colleagues referenced a few hours ago, there was concern that these like FIASP and current ultra rapid analogs are not rapid enough to impact on time and range with fully automated closed loop systems.
Okay.
And it might be that the enormous progress of these technologies will motivate development of truly ultra rapid zinc-free insulin formulations, taking advantage of this intrinsic stability in order to achieve the metrics that are required to make the time and range above 90%. And as an aside, with respect to glucose responsive insulins, most of the technologies discussed today pertain to intermediate acting or long acting glucose responsive insulins, which are a fantastic advance. But wouldn't it also be wonderful to have an ultra rapid glucose responsive insulin within a fully automated closed loop system to get the time and range above 90%? So that's our dream.
Yeah. But that's the holy grail we are looking for exactly with this symposium. So thank you very much. If there are no further questions, just a recommendation which you can read in the chat later on. And I can only thank you very much for this very clear, straightforward presentation and report on your activities and reflecting your ambitions in this field. Thank you very much.
Thank you.
I think I have to hand over now to, is it David?
Yes, Reinhard, it is. So hi, I'm David Kelley. It's my pleasure to introduce our next speaker, who is Dr. Alborz Mahdavi, who will be speaking on glucose sensing insulin. As background, he is the founder of Protomer Technologies, which focused, amongst other things, but focused on glucose sensing insulins. That company was acquired wholly by Lilly in 2021, and Dr. Mahdavi continues as a vice president at Lilly to oversee the line of research and drug development started at Protomer. As background, he has lineage from the Bob Langer group that we've heard described for several of the speakers and heard from Dr. Langer himself at MIT, and subsequent to that did research with David Terrell's mentorship at Caltech, for which he received a number of awards, including from the JDRF, and that gave impetus to his founding of Protomer Technology. So without further ado, we look forward to your talk on glucose sensing insulins.
Great. Thank you, David. It's nice to see a lot of friends on this call. And what I thought I would do today is talk more generally about glucose sensing insulins and what may be interest to the team that already has a lot of knowledge about this area, and go through some of the history of Protomer as it pertains to a company that works in glucose sensing insulins, and now as a wholly owned subsidiary of Lilly. So let me share my screen, and please let me know if you guys are able to see my screen.
Yep, we can see that fine. Thank you.
Okay, great. So as mentioned, I will go sort of briefly through some of the efforts that we had at Protomer in terms of developing really a company that works in glucose sensing insulins. And that is not a trivial task. As you can imagine, when we started Protomer, and here is a sort of a timeline of the company, it took some time for us to be able to actually get investments to support the work that we're doing. So here is a kind of a quick timeline of Protomer. We started the company, I started the company in 2014. This was really supported from day one by Juvenile Diabetes Research Foundation, JDRF. I initially got an award from JDRF for the Glucose Responsive Insulin Prize, and subsequently wrote several applications for grants to the foundation, and eventually was successful in getting support initially from JDRF. But during that time, we also had an opportunity to connect with Sanofi, and had a collaboration with Sanofi in this program from early on. And the idea there was to explore some of the concepts that I'll talk about today in glucose sensing insulins. Some of those early concepts didn't really weren't sort of ready for prime time, but over the years we were able to develop these concepts a little bit further along and make quite a bit of progress, really starting in 2015, understanding what are the things that can work in glucose sensing insulins. And later on, we were fortunate to win a couple of sort of prizes. One of these you can see here was by T1D Exchange for an idea that I'll talk about in a little bit. And then in 2019, Eli Lilly invested in Protomer, and subsequently acquired Protomer in 2021. So one of the things I did want to mention in the talk is that it is great to develop these ideas at sort of, if you will, in academic settings and scientific investigations of what mechanisms may work. But it was a big challenge to translate that into sort of a company that can get investments. And receiving investments, sort of equity investments in this field, can be challenging. So the support of groups like JDRF and the support that you guys provide to early stage companies is critical to be able to actually work in this field. It's not sort of the typical startup or biotech startup that you see that oftentimes work on programs that will have a 5-year sort of turnaround time. As you see in most oncology programs, it's a very fast timeline. And working on glucose sensing insulins as a very challenging field is not the easiest value proposition for most venture capitalists. So I think that's an important distinction in this field versus many other fields where we have development of concepts towards the clinic. So just very briefly, when I started Protomer, obviously you can see some of my board members on top. We were fortunate to have Katie Elias from the JDRF T1D Fund be on our board, and also Tim Dolan, who was representing Eli Lilly after the investment that Lilly made in Protomer in 2019. I also have our scientific advisory board members on the bottom. So Dave Terrell and Mikhail Shapiro, both at Caltech, really helped us in sort of guiding the company in terms of early days. And then Brad Pantuluti at MIT, obviously being very relevant in terms of the technologies that he works on and the technologies that we work on at Protomer. So what I didn't want to do today was I didn't want to go through a detailed discussion of what looking at structures and the chemistries and the insulins that Protomer works on, but really talk about sort of bigger picture. Where is the field at, and where are we headed, and what should we be aiming for in glucose sensing insulins in terms of realistic sort of goals for this field. It's been a very challenging field for a number of reasons, which I'll talk about, and I'm sure was mentioned also today. And a number of my friends who are on the call have also struggled with these challenges. I got into the field naively, not realizing some of these challenges early on, but over the years, we've been fortunate to work on some of these. So one thing that I did want to mention, I know that the talk is titled Glucose Sensing Insulins, but it's also important to recognize that glucose sensing as a modality is actually sort of a larger idea, one that can be applied to other areas. So one of the things that we investigated early on was this idea of a smart glucagon. So a glucose sensing approach that's applied to glucagon instead of insulin. And this is an idea that's, has been near and dear to my heart because it is an interesting concept. So that it's really the reverse of what you do with insulin. A glucose sensing insulin turns on at high glucose. But if you have the ability to sense glucose, then you can also engineer glucose sensing glucagons that will turn on at low glucose. And so really as an augmentation to existing insulin therapies, but as a way to reduce hypoglycemia. So the idea here really is if you could develop this in the context of a molecule that would be ideally a once weekly smart glucagon, that's really, that's 52 injections a week and eliminate all hypos, and then allow people to be more deliberate with their insulin injections, not fearing hypos. I think at the extreme case, this program can also enable fixed dose injectors on insulins, knowing that you have coverage on the hypos. So again, an area that hasn't necessarily gotten as much attention as glucose sensing insulins, but one that we think has a lot of value and be interested to also hear from any investigators who are working in this area. So maybe as a plug for our work at Lilly, one of the things I want to mention is that we are obviously always interested in looking at innovative concepts at Lilly. Obviously, we're excited about the work that we're doing at Protomer in glucose sensing insulins and these glucose sensing programs. But we remain open-minded and always looking for interesting concepts that come our way. So a quick overview of where Protomer is at today. We've moved into a brand new building in Pasadena, right near Caltech. As you can see on the picture on the bottom, we are putting together really interesting, innovative programs with Caltech, making sure that the synergies are maintained. You can see on the top right, the CEO of Lilly, Dave Ricks, at the grand opening of the new building, and Ruth Germano, the head of the diabetes group at Lilly. And then on the bottom, you can see some of our team members. And on the very right, Dan Skrzywanski, the CSO of Lilly. So we are fortunate to be a part of Lilly. And as part of that, I wanted to just go through some of the ways that we think about glucose sensing at Lilly. Hopefully this will be helpful to others who are on the call today, and also maybe motivate some discussions with the team. So really the foundation of what we work on is, improving and simplifying glucose control. And this is really kind of the foundation of glucose sensing insulins in a way. The idea is, how do we really make insulin therapy easier for people? And today, the state of the art is you have an insulin pump, you have a CGM, and all of the devices as well as the algorithms required to maintain glycemic control. Of course, the really sort of the transformative part of this would be if you had an injectable insulin that could be dosed a lot less frequently, and the insulin was glucose sensing enough that it could give you coverage. And this really is a continuum of innovation, and I'll talk about that. But that would be the transformative idea here for glucose sensing insulins, the ability to inject insulin without fearing hypos, not having to measure units of insulin or count carbs the same way as we've done so far. Ideally, maybe even dosages that are a little bit more fixed, and really simplifying insulin therapy. So that's what we're interested at Lilly. And in terms of therapies or anything we look at, whether it's internal or external, it will have to be a simplifying modality. So maybe just a couple of points of why glucose sensing insulins are really difficult to make, and what makes this a challenging field. I really like this plot on the left because I think it illustrates the concept quite well. So if you look at glucose levels in the blood and the changes in the concentration of glucose in the blood, what you see is maybe a 1.5-fold in change in blood glucose concentrations between meals, and so you see maybe a glucose change from 4 millimolar up to 6.5 millimolar or 6 millimolar of glucose at mealtime. And this is obviously in a healthy body, but obviously for diabetics, these numbers will fluctuate quite a bit more. But what we see physiologically for serum insulin levels at these glucose levels is the serum insulin levels are changing more than the glucose amount. So if the glucose amount is changing 1.5-fold, we typically see the serum insulin levels change almost 3-fold in response to those glucose fluctuations physiologically. And so why is this challenging? The challenge is any sort of sensing modality you would have for glucose in the context of a glucose sensing modality will have to counter this sort of linear effect of a 1.5-fold change in glucose responding to a 3-fold change in insulin levels. So if you really want to have a successful glucose sensing insulin program, you have to engineer your approach in such a way that the fold changes in insulin activity or release, if it's a releasable system, are more than the fold changes in glucose levels in the blood. And from a molecular engineering perspective, that's obviously a very difficult thing to achieve. And that's historically been one of the challenges. The other challenge, which I think is illustrated by the picture on the right, is if you actually calculate how much soluble glucose is circulating in the blood at any given point, and you measure, what's the concentration of glucose, you look at the volume of blood, there's about 5 to 6 grams of soluble glucose circulating in our blood at any given point. And so if we have an ice cream that has 40 or 50 grams of sugar going into our system, you can imagine what an exquisite control system that we have in our body that it ensures that we only have 5 grams of glucose circulating and the rest of the glucose is removed by the action of insulin. So this sort of illustrates the challenge of developing glucose sensing insulins, both on the sensing part of being able to be responsive to physiological levels of glucose, but also on not overactivating and causing hypoglycemia. So I think everyone here is fairly familiar with the action of insulin and the different types of insulin. So I'll skip this slide and go to the next slide. When we really think about the innovation curve of glucose sensing insulins, we really think about this continuum. So we've developed, in the past, ultra-rapid mealtime insulins. We've developed connected care modalities. We have weekly insulins in the clinic now. So what is next? What is in the innovation curve of insulins? So we think that probably the lowest case is glucose sensing insulins that replace mealtime. So the idea here is really these are injected at mealtime, and the main sort of value proposition here is no mealtime math. So really a simpler glucose sensing insulin that's provides mealtime coverage. You still have a basal glucose that's being taken care of by a basal insulin, but you have these injectors of your glucose sensing insulins that are injected at mealtime and really a much more flexible approach. Now, I think Michael Weiss made a comment on this just in his talk earlier about the possibility of having these in pump formats as well, and that's certainly true. That if you had glucose sensing insulins for mealtime, that could also be applicable to pump-based systems. So we think this is probably the lowest case in terms of the first in the innovation curve. Now, of course, at some point, glucose sensing insulins that cover basal insulin needs would be interesting as well. So these types of insulins would be replacing the sort of the basal long-acting insulins that we would have. And the idea is you have coverage of the basal glucose, but maybe some coverage of the mealtime postprandial glucose needs as well. And so again, in the innovation curve, this is probably would be the next frontier in terms of glucose sensing insulins. And then over time, we would have more simplified, possibly maybe even before the basals, more simplified mealtime insulins that have no math, and then maybe like a single injection. And then over time, probably ones that cover both basal and mealtime. So again, it really depends on the modality that's utilized by the insulin as well. And so I guess that one of the themes here to mention that's really important is this may be a situation where one type of approach doesn't address all of these different categories. And so it's not a one fit for all of these problems, but maybe there are different approaches that would be applicable to different types of problems that we were going to see in this innovation curve. And so, as I'm sure, as all of you realize, the different approaches that you see today may be applicable to different parts of this innovation curve. And again, the sequential nature of this curve is not as important as just realizing that there is variations here. And so obviously, the ideal scenario would be a full control. We have a glucose sensing insulin that really covers both mealtime, postprandial glucose needs, as well as the basal glucose needs and coverage in all of those scenarios. Obviously, a very, very high bar for glucose sensing insulin, and with our estimate, it will take many generations of glucose sensing insulins to get to this scenario. So what are the main challenges that we see in glucose sensing insulin field? And these challenges really, I'd say they're in 4 categories. And these are in order of, I guess, challenge. So really the first one is developing glucose binders that can respond to physiological levels of glucose. And this is not as trivial as it seems. If you make any high affinity binders in most other contexts, they would be fully saturated at blood glucose levels of millimolar range of glucose. So how to develop sensing modalities that can sense changes in millimolar levels of a soluble small molecule, such as glucose. The second part is really how to control insulin activity using these glucose sensors. And there are a number of different approaches that are used, which I'll talk about in a bit. But really, it's one thing to be able to bind to glucose. It's another thing to be able to control activity of insulin in response to that binding. So we know a number of different sensors that have been developed by a number of really innovative investigators where you can actually measure glucose levels by the binding to that sensor using maybe spectroscopic or other modalities. But that's not the same as controlling insulin activity. So that second part is a very non-trivial challenge. And then the third part is making sure that you actually develop this in a context of a molecule that has enough potency that you can actually have a relevant injection dose with regards to potency of the molecule. And then the last one, and not the least, certainly, is that you can make such a compound at a low enough cost that within the sort of the context of insulin pricing, it actually makes sense from a cost of goods perspective. So how do you develop these concepts in such a way that they're cost effective to make? And as I'm sure you know, you need to make metric tons of insulin every year for the use of these concepts. So any idea that would involve novel chemistry or exotic methodology probably would not be a viable option from a commercial perspective. So I'm going to use a couple of concepts from this really great review article cited on the bottom, just to kind of cover some of the approaches that people have used today that, what may or may not be interesting within these contexts. So on the left, you see a lot of the sort of the non-reversible sort of approaches. These are sort of agglomerated technologies, a number of these sort of gel-based systems or release mechanisms where binding of glucose to a sensor allows the release of insulin in response to that binding. And then the released insulin is active in the body. So one of the challenges with these non-reversible systems has always been the safety, right? If you release too much insulin, that can be, quite problematic from a safety perspective, leading to hypoglycemia. So how do you really reduce that that risk? And that's where we see the reversible systems having a lot more applicability in terms of being a safe enough system to be used in glucose sensing insulin. These really fall into a couple of categories. There is the chain locking mechanisms on the top right shown here. So the idea is if you can change the conformation of insulin, you can control the activity of the molecule in response to glucose. At high glucose, you get the opening of the chains and the activation of the molecule. There is the insulin carbohydrate conjugates. The idea here is the coherence of the molecule is really controlled by the presence of glucose. At high glucose, you'll have a slower clearance of the molecule and higher systemic exposure to that insulin. And then there's the albumin binding or human serum albumin binding methodologies where there is some sort of reversible binding to albumin, and that's really controlled by the level of glucose. Again, we think these reversible approaches are a lot safer and probably more viable as a modality. The other one not mentioned here, but I have in later slides, is binding to glucose transporters is another modality that really early on, there are a couple of groups, Gu's lab as well as Sanofi investigated this approach early on and published on it. So the reversible glucose sensing insulins are exciting because we know that these systems can be safer. We know that the risk of hypoglycemia can be minimized. And the reversibility of activation really is the key here in making sure that, that even if there is a high activation of this insulin, that you have shut-off mechanisms. And so the way that at Protomer we've pursued this without going into all the details is looking at really conformational control of this insulin, going from a inactive state to an active state in response to glucose, and then being able to do that in a reversible manner. And these, obviously these concepts that we pursued early on have also advanced and evolved into, even I would say early on from 2015 into more sort of other modalities that are still fully reversible, where we think that the safety mechanisms are there. So maybe I'll skip this one and just say the way that we approach this problem from really early on was instead of designing these systems from the ground up and coming up with a design, what we really did was we made search, if you will, technologies where we could screen through many variations of insulin, modified insulins. And so we spent several years at Protomer putting together the platform technology to be able to do this. The platform technology, which we've discussed briefly here, is called molecular evolution of peptide sensors. The idea is to make very large libraries of these molecules where we know that the glucose sensing modality is incorporated into the molecules, and then search through those millions of variations of the molecule to really identify variants that can respond to physiological glucose levels, and then further develop these through study activity relationships and SAR studies. So maybe just in kind of concluding with some guiding lessons that we've learned over the years in terms of glucose sensing insulins. I've bucketed these into sort of 5 different approaches. And the key word here is the linear versus the nonlinear methodologies. So, I would say with the glucose transporter systems, those could be in either a linear or nonlinear category. But the idea really there is that it's a releasable system. And what you're really aiming for here is, are we looking at competitive binding or non-competitive binding? I think for various reasons, which I guess I won't mention for the sake of time today, it didn't seem that this necessarily was an idea that could be advanced to lead to those sort of nonlinear responses. Sanofi had an early program in this. They have published on it. They have patents on it. And, you know, again, looking at the data that's generated, we also certainly have internal efforts. This one is one where the mode of activation would be important to consider. In terms of other sort of, you know, systems, if you look at linear systems, there is the idea of developing a molecule that binds to an insulin that has a some sort of a glucose sensor on it, and then the binding of glucose to that portion of the insulin then releases the bound molecule. This was a concept that was developed by a company, amongst others. This company was called Glycostasis, which Lilly acquired early on and worked on. And so the idea again here is really understanding, you know, that binding will be a linear phenomena, which also will potentially limit the applicability of that overall approach. Now, of course, you could make this a nonlinear approach by sort of the third modality, which is binding to an insulin that is different than where the glucose sensing is done on the insulin and then released in response to that binding. So that is a very interesting concept, and one that is, from a molecular engineering perspective, not a trivial one to do. One that hasn't been talked about a lot, but we think has quite a bit of value. And then the fourth and fifth, which I think I've mentioned today, really these sort of releasable based systems, whether these are bound to glucose and cause the release of the insulin or change of insulin, or there is some sort of human serum albumin interaction that, in addition to the release of the insulin, can also provide modes for sensing. So in terms of future directions, you know, where are we headed with glucose sensing in general? So obviously glucose sensing insulin is interesting in itself. We think there may be, and we would be interested if there are glucose sensing insulins that are made that have preferential hepatic effects. We think those would be some of the next frontiers in terms of glucose sensing. I certainly want to mention that, you know, as my role as a VP at Lilly now, I'm sort of in charge of making sure that we are making good progress in this front. And so if there are interesting concepts and ideas that the team here or others that you know of are working on, we would certainly be interested to look at those as well. Glucose sensing insulins with complete off switch at hypo would be of interest as well. And the idea here is that you essentially completely eliminate the insulin that's on board at hypo and then reset the system. So that really would be a complete hypo, a complete turn off at hypos would be very interesting. Pump specific glucose sensing insulins, I think we talked about that. And then lastly, I do want to just mention that glucose sensing glucagons would be of value as well, and certainly one that's not the topic of discussion for this talk, but one that's of very high interest to us, and certainly would be delighted to connect with anyone who's working on that as well. So I'll stop there for any questions, and I'll stop sharing the slides as well, and happy to take any questions.
Well, great. Well, thank you. Thank you, Mahdavi, for a very nice and clear talk. And I really enjoyed the way you began by framing it out in terms of the 4 grams of circulating glucose and how to build a signal that translates to insulin action that is manipulated by relatively modest changes in the mass of glucose circulating. And I would think the challenge, I'd like to hear your thoughts, because a couple of times you called out glucose responsive glucagon. And to me, that amplification calculus would be even more challenging, because you're symptomatically, one would become hypoglycemic with a 1/4, 1/3 reduction in the volume of circulating glucose, and yet one needs to amplify glucagon action 5-fold to really formulate a bona fide counter-regulatory glucagon response. So How do you tackle that, either chemically or by mass of inert glucagon circulating, or how have you thought about solving that mathematics?
Yeah, that's a really great question. Glucagon is, like you said, this is not the one that people typically think about, right? So glucagon is a very different molecule. You have two levels of challenge. One is how much glucagon how much glucagon activity do you have, especially in type 1s, where you have the beta cell response that's gone, and then the alpha cell response can also be compromised over time. So to what extent can you recover that response? I think that's an important thing to think through. But mechanistically, for developing a glucose sensing glucagon, I think the same glucose sensing approaches that we have developed for insulin, at least in our hands, are directly applicable to glucagon. The challenge really is, I think, more so from a perspective of, if you think about from a patient's perspective, most of the time they're not seeing the immediate action of that drug because it's in the background. And if you have a glucose sensing glucagon, typically with insulin, when you inject it, you see the effect right away. And you say, okay, I know why I'm taking that insulin. With glucagon, if it's a once weekly injection of a glucose sensing glucagon, hopefully you never actually see that effect because you never go into a hypo. And so I think that's just more of a conceptual, block in terms of, why do I have to pay for this medicine, or why do I even have to cover this medicine from, but we think there's a lot of value there, because the other side of it is that it gives you a lot more flexibility with your insulin. And I'm not sure if I'm answering your question. I think if your question is, how do you make that conceptually? How do you make that glucagon? That's a much longer discussion, right? But I'd say just generally, if your question is more, how do you develop glucose sensing glucagons? The exact same glucose sensing modalities that we use can be used there. You just have to, with the sensing, you have to tune the sensing so that your sensing is not done at the hyperglycemic state where you activate the insulin. Your sensing is done at the hypoglycemic state at much lower glucose levels, where you actually have the turning on in the reverse way for glucagon. And that really depends on how you design your glucose sensing insulin. So, for example, if you have the glucose transporters, just taken as an example, that could be challenging to do with glucose transporters. But the other modalities that we mentioned, certainly we think there is a good possibility of approaches there for glucagon.
Great. Well, thank you. I don't know if we have time for one more question. It looks to me, unfortunately, that we transition to the next speaker. So thank you again for a great talk. Professor Michael Strano, whose talk will be, and it's the last formal talk before we go into broader discussions, but his talk is entitled Diabetes Therapeutics from a Computational Design. And let me introduce Professor Strano. He's currently the Carbon P. Dubs Professor in Chemical Engineering at MIT. And he's a postdoctoral fellow at Rice in departments of chemistry and physics under a Nobel laureate there. And it's climbed the academic ladder initially at University of Illinois at Urbana-Champaign before moving to MIT. And his research focuses on the area of biomolecular nanoparticle interactions and the surface chemistry of low-dimensional systems. So, Michael, welcome to today's symposium. We look forward to your talk.
Good. Well, thank you. It's my pleasure to be here today, and this is exactly the community I would like to talk to and learn from. I can teach a little bit, but I need to learn from this community. They have what I'm going to talk about today are in silico simulation models of the glucoregulatory systems, and I have one take-home message, and it's that it's that it's these computational models that can model the human body and model animals and the glucoregulatory system. They have evolved and they have gotten accurate enough to where now we can make predictions of experiments. We can predict the efficacy of their therapeutics. This has been a slow process through the decades, but that's my take home message, not just from contributions of my group, but really as a field as a whole, that computational models of the glucoregulatory system have gotten refined enough to where we really can make predictions. And it's not a computational hardware innovation. It's not an AI or chat GPT. It's not machine learning. It's just really careful, good medicine, good data collection from many of you, great math and biochemistry, and just it has come and it's gotten to the state. So that's what I want to convince you of today. My group, I lead a research group of about 20 people here at MIT. I would say about a fourth of our work is on diabetes, and there's a portion of that that's just thinking deeply about humans. And our claim to fame is that we actually have a very accurate model of not just the human glucoregulatory system, but it can describe animals, and it can describe the intersection between those two. So we can take results from humans and predict what animals would show and vice versa. And I hope to convince you of that today. Alborz did a great job of summarizing what a GRI is. I don't think I have to summarize that for this community, but this idea of a therapeutic or insulin particularly that becomes active or becomes dormant based on some therapeutic endpoint, in this case glucose, I think is a revolutionary concept in medicine. From a computational standpoint, this drug becomes a line of code, right? It's like a small machine, and I can describe it with a mathematical equation, sort of like if this much glucose, this much potency. It's a sensor and it's an actuator, and that makes a glucoregulatory model of the human body very, very valuable. What will be the physiological response to this drug? So there's what we call a GRI, is a therapeutic that acts in this way. There's also very innovative ideas that we just heard from as well on insulin delivery systems that also act as a GRI. So it can be a patch, or it can be a particle that releases insulin in response to glucose. And this is exactly the community to have that does not really need a background in that. I'm not going into math. I'm skipping all the math. If you're part of the computational space, happy to have a sidebar with you offline. I've kept the math in this talk very, very minimal. These are pharmacokinetic models. Researchers working in bioinformatics or working on computation will be familiar with them. They divide up, say, the human body and the organ systems into compartments, and they parameterize the mass transfer, and chemical reactions that occur inside the compartments. They're all connected through the circulatory system, and so you can put a therapeutic into the simulation, and it outputs in time what happens to the therapeutic as it moves through these compartments. It produces a large parameter space, a large number of parameters, but through the years, we've been able to use those parameters to train models. It's not just the work of one group, it's really the work of an entire field. So hopefully, and if you have questions on the math, it's not particularly difficult math, but it's just large systems of equations so when we evaluate a therapeutic like a GRI, it's important to define in mathematical terms what is good, what is efficacious. Okay, and so here for diabetes, just talking to clinicians, we've learned from you, we have a very simple cost function. We have a hypoglycemic risk score, and we have a hyperglycemic risk score. And it's literally if we were to plot the glucose concentration in the human or animal, the first just penalizes the downward excursions in the hypoglycemic range, and the hyperglycemic does the same thing, and we integrate that. So there's a temporal penalty, and that gives you a score. It's a great way to evaluate whether one therapeutic like this GRI is more effective than another. And what you get is a drug design space, which we think or hope will be very valuable to this community and people thinking along these lines. Now, I can explain this. This is for one particular kind of GRI, but it maps this penalty function in this case in terms of two variables, right? So there's drug design parameter one is how fast does the GRI activate and become potent in response to insulin, and in this particular case, the drug design parameter two along the x-axis is just the glucose binding equilibrium constant, or 1 over the dissociation constant. And what you get is this design space, very useful. So in the blue, you will get a physiological, you'll get an efficacy that's within your design space. And in the red is a GRI that will fail. It will not be able to control blood sugar the way that we want. And this constitutes a design space. The medicinal chemist it's not a knob that they can turn, but they can definitely measure parameter one and parameter two. They can definitely take a therapeutic and put it on this map. And they have limited ability to control. Those of you who have thought about glucose binding and so forth, you know you have some control in terms of chemistry. So we like to think of this as a design tool. So if I add a third dimension in the z-axis, hopefully these are not too complicated, but you get a three-dimensional space if you add dose. So this three-dimensional graph is the same as what I showed. It's along the x and y are the drug design space, but then dosing actually matters. For the GRI, the dose defines the performance as well. So you get this efficacious volume. And so these calculations can also predict how dosing of your construct will affect its performance. And so what I've mapped out here, this is the three-dimensional space where we expect to be able to regulate glucose from a given GRI. So you have zero hyperglycemic risk and zero hypoglycemic risk. So this gives you when I say design space, this is what I mean conceptually. And you can ask all kinds of questions. We have a computational model. Our claim to fame at MIT is we have a computational model that's very accurate for rodent models. We have done mini pig, and we're in the process of doing canine. Our team is fairly taxed, but what this means now, and also humans, from human data, and what that means is we can take animal experiments and we can predict how they will map to efficacy in humans, and vice versa. And there's lots of questions that you can ask. So just to illustrate this is this three-dimensional design space. This is the intersection of rats and humans. And you see this bright magenta, that's the region where that therapeutic will be successful in rats and also successful in humans. And we have mice and humans in the center, mice and rats, and then you can do all three. You can do humans, mice, and rats, and it's an even narrower space. And this brings up a lot of questions and a point that I'll make a little bit later. But successful drug designs in some animal models won't translate to humans. There's lots of questions that you can ask, and I'll make that point a little bit later. Okay, but these models do very, very well in predicting experimental data. This is actually data from my colleague who I think is present here, Michael Weiss. We've had a very fruitful collaboration with the Weiss group. They're absolutely fantastic scientists and medicinal chemists. This is for healthy rats, a population of order of n equals 10, but this is a subcutaneous injection. So it's a routinely done experiment. You're going to inject insulin subcutaneously, and you look at the resulting glucose response, and this computational model for rats does a very good job of matching and predicting the response for healthy rats and for the diabetic rats. It gets the dosing very, very well. It gets the dynamics well. On the right is actually data from my colleague also here at MIT, Dan Anderson, and this is data right from his literature study, and these are also healthy and diabetic mice. And from these experiments, we can get high fidelity computational models, and then we can ask if we take an arbitrary GRI and we plug it in to those animals, we can ask what will be the resulting performance, things that we can measure. So these models have gotten to be very high fidelity, very high accuracy. And this is an example of this is a glucose clamp. So this is an experiment done, I'm sure many of you are familiar with it, where you clamp the glucose level, you keep it constant, and then you will inject the therapeutic, or you'll inject insulin, and then you'll keep track of the mass balance, how much glucose has to maintain the clamp from control theory. So this is actually for canines. This is Merck's data for MK2640. I'm going to talk about that a little bit later. But you're supposed to see here that this happens to be for canines, and we're in the process of doing canines now. But you can see here that the simulations themselves do a very good job of matching the clamp experiment. You're supposed to see that there are successful trends here. On the left is MK2640. On the right is actually just regular human insulin. So you're supposed to see the comparison between the two. And you see here that their GRI is modulating, but their insulin is not. So that's supposed to be their control. Okay. So now there's lots of interesting questions that we can ask. We can ask. This space brings up a lot of questions. So first, the expected space is where your animal model successfully predicts therapeutic efficacy, and it maps to humans. But there's lots of other spaces. There's this space of false positive. This is and it's a fairly wide. So here I show for mice. These are spaces where the therapeutic is predicted to be successful in mice data, but will be unsuccessful in humans. This white space is just true negatives. Will fail always. But then we have this disturbing false negatives. This space means a therapeutic that at least rats and mice would show is not efficacious, but will work in humans. And we have to think about how we'll handle the regulatory process. I think we will need a lot more confidence in models like this to really explore therapeutics in this space, but I think that's coming. I think it's coming, but it won't rely just on models in the current form. So there's lots of questions that you can ask, and we've been applying this tool. This tool is very valuable for applying for drug design, for evaluating data in the literature. There's really a gold mine of data in the literature. This is a paper that we have under. This is a study that we have. That's under review now. This is Merck has, I think, the only GRI to go to clinical trial. It was a very ingenious GRI. It was based on applying two saccharides to insulin, which would then bind and compete with the mannose receptor. So the idea here is it's a clearance-based GRI pathway where this therapeutic under hypoglycemic conditions would bind to the mannose receptor and just be cleared, but at high glucose would compete with the GRI, which would release the GRI out and allow it to have activity. And it's an ingenious pathway. Its efficacy in humans, though, didn't reach the level to where the clinical trial was considered successful. So this tool can help us all understand why. What I want to convince you of, though, is that you can take a drug and make it into equations very accurately. So this is their GRI. You can break this ingenious mechanism into just a receptor binding of the GRI to the mannose receptor. You can the clearance rate, and then the competing glucose binding to the receptor. You add those together, you get an equation. It fits their in vitro data really well, right? So it can describes their therapeutic. Now you take that equation, just put it right into the simulation, and what do you find? Well, we do a very good job of being able to predict their clinical data. This is the black points are what we're simulating, and the boxes are their clinical trial data. This is for regular human insulin. This is their GRI. We're able to match the results very, very well. This is for humans, and then they have animal data for mini pig. Well, this is interesting here. This is if we take their animal data and we simulate humans, we actually predict a very lackluster lackluster clearance level. If you their dog experiments, however, showed a much larger 25%, and this was very encouraging. And we but so our simulation can take their dog experiments and predict that the result in humans would have been less efficacious. And if you want a test of that, we actually can simulate in mini pig. And if you simulate in mini pig, we would have predicted that you would get even more encouraging results, but that still you're going to have issues in humans, which is what we care about. So there are some takeaways here. So first, if we take the clearance rate from the mini pig, we can now dissect and find out what's wrong with this therapeutic, or where should you direct your attention. If we take the mini pig clearance rate and we put it into humans, you still get a predicted GRI potency that is less efficacious. So that's what's shown here. We're sort of making a virtual Frankenstein. You could take this from mini pig and put it into humans and ask. So it's useful for mechanistic deconstruction. Well, if we substitute the mini pig mannose receptor distributions and put that into humans, you get a much bigger clearance change. So really, it's this mannose receptor distribution, and I think the upside of that is you want to design a therapeutic around that. And then, because what ultimately is happening in the Merck case is the therapeutic range in humans is shifted to higher glucose concentration. And it's shifting to a range where it doesn't help as much. If there's a way to shift that back, when in going from animal to human, then you'll recover a very large clearance rate. This 33% is a big enough change for the trial to be successful. So anyway, it's just an example to show you how you can piece this together. We have studies now of different GRI constructs. I'll make sure I'm not going to go over. We have class 1, class 2, and class 3. We're just taking GRI ideas from the literature, and we're trying to simulate them. We're trying to help researchers as much as we can on where to look. A class 1 GRI we heard about, that's where the molecule itself is responding to glucose. It's becoming more potent or not, but it's in equilibrium. Okay. A class 2 are the therapeutics, like a particle, like a cage, and glucose unlocks the cage, and that's what our previous speaker called. It can be irreversible, but I mean, it can be reversible too, but there are versions of that are irreversible. And class 3 is just a device, like a microneedle array. It's instead of circulating around the circulatory system, it's at one compartment, and it's still going to act as a GRI, but it's in one location and in one compartment. And we can ask the same question. We can say, where is the efficacy space in humans, in rats, in humans and rats, but now we can do that for each class and come up with some design rules. And I don't want to go through all of this here. I just want you to know that these exist. And if you're working on these, we want to help you. We want to give you advice, because the criteria for making a successful GRI, it depends on those classes. So in class 1, this K forward, K reverse, the ratio is the equilibrium constant, and so both are very important to get a successful GRI. Class 2, though, what you can see here is that it's really just this forward rate. Right, you can have a lot of this equilibrium binding. That the situation is very tolerant of it. It's really, you should spend your focus on the forward rate, and it's the reverse in class 3. It's counterintuitive, but here you want to really focus on receptor binding of glucose, and there's a region where you're tolerant of how fast, and that's because it's already in a compartment. And so you have the transport there. So anyway, hopefully I've started to convince you that in silico modeling, useful tool, and that we're now at the stage where we can predict and we can help in drug discovery and drug design. And I'm very pleased to work with my colleague Michael Weiss and others in the field who have ideas and want to test them out in silico. So my goal is to make sure in the near term, we're doing work now on glucagon, we're expanding these models to different animal models. We're focusing on canine, and ultimately we can have a tool that can also help in animal experiment design. Ultimately, these tools can also, they can also focus and extract more information from animal experiments, making them smaller, less expensive, and yield more information and speed up and accelerate the innovation process. That's our goal. So so with that, I will just thank NIH, support from JDRF, and also the Helmsley Charitable Trust. And I'd like to thank the audience for your attention. Yeah, thank you.
Dr. Strano, thank you. Thank you very much for your talk. It was very interesting to hear about this model. I have to confess, on a personal note, as someone who was deeply involved in the discovery efforts around NK2640, one of the issues we struggled with was trying to predict the cross-species translation of the mannose clearance system, and we could never put our hands around that in a quantitative way and had to do it empirically and suffered the painful consequence. So I won't torture you or the other listeners with that tragedy. But how sensitive is your model to the issue of insulin resistance? It may not be that pertinent for the pure drug development in a conceptual way, but just as an in silico model of glucose insulin homeostasis, how do you factor in mathematically variance in insulin sensitivity?
It's an excellent question, and I'm not going to suppose that I'm the expert there. I have started to interact. I've started to ask that question of my colleagues in the medical community here in the Cambridge area, and I need to learn more about that, and I need to learn from this community. And so insulin resistance can, it's of course it's a variable from individual to individual, but it also apparently it can also change throughout the day, and it can change with activity. And there, in my opinion, there needs to be more thought on that, how to quantify it, how to extract it from data. I think there are ways to do so, but I believe with models like this, we're at the start of understanding that, and so it's a great question.
Great. Let me see if there are any questions in the chat room. So here's one. Did you perform the simulation for humans prior to the clinical study or thereafter?
No, not prior to the clinical study. No. This is all of our. We started on this effort. Yeah, we came afterwards, and yeah. So this is a retroactive evaluation.
Oh, if this is in reference to the 2640, yeah, this would have been done retrospectively.
Yes.
Yeah, unfortunately, we didn't have your insights prospectively.
Right, and also these tools. I think if you go back in time, these tools were not ready yet. And again, it's not just my own work, but it's been the work of the entire field, including just gathering data. I think confidence in these models is growing. Yes, but the way we construct our paper, you can ask the question, given this information, can you predict the outcome? That is our study. So in theory, tools like this in the future can be very good guidance for clinical trials, right? And mainly for extracting and refining what you can conclude from animal studies. Yeah.
Great. I think since we're moving into discussion, there is one more question in the chat room. And so let me take the liberty to pose this one to you, Professor Strano, and then we'll move to the broader discussion with all the speakers. So this question comes from Tim Heise, one of our co-chairs, and he asked, how important are pharmacological properties, i.e. time of onset of action, duration of action for the mathematical model?
Yeah, that's exactly, yeah. So time of onset, I know I went through it quickly. Time of onset is that K1 or K4 forward. And you see that in some cases it's critical. If the medicinal chemist has that as a knob and can tune it, well, then you can make light work of this problem. It's and there there are other things that are harder to put in mathematical form. Clearance matters quite a bit, and we have to understand that empirically. And so these are good questions. But the one that your intuition about the delay and onset of going to an active form, that is critical. The timing of that, that has to occur within a window, and that seems to make the difference between a failed therapeutic and a successful one.
Okay, well, thank you. I don't see any other questions in the chat room. So thank you again for your talk. And Tim, I'm going to turn it back to you to moderate the discussion panel.
Thank you very much, Michael. And a big thank to all, excuse me, to all the speakers for excellent presentations. I really enjoyed this afternoon, and I look forward to discussion. So to all those in the audience, just to remind you, this is basically your last chance to ask questions to the speakers, either to one speaker, and if so, please just indicate which speaker you would like to address the question, or of course, question also to all the speakers. And if there are questions from my co-chairs, they are of course happy to chime in too. And I thought I start off basically a bit where the last presentation ended. I mean, it's great to have mathematical models, and it's even greater to see all these different fascinating approaches and actually very promising data, often in rats or dogs or mini pigs. But still, we only have one published study on a glucose-sensitive insulin in humans. So I wonder, what do we need to really move on to clinical studies? And I perhaps start with Dr. Gu as one of the first speakers who has presented so elegant approaches.
Hi, Tim. Yeah. So actually, as I mentioned in my slides, we just initiated the clinical trial, the IIT test of the glucose responsive microneedle patch, especially including the the safety evaluation as well as the glucose responsive assay. Yeah. So we are working on this currently now. And meanwhile, in the end of my talk, I mentioned the conjugation of the insulin and the glucose transporter inhibitor. And currently, we are also preparing for the IIT test. The only thing is like we need certain time to synthesize sufficient compounds. Yeah.
Okay, thank you. And Dr. Strano, do you, if I understood you correctly, you could basically try to model the preclinical data to human data, so that would hopefully speed up the development process quite a bit.
Yes, and if I could make a request, I don't know how, but if the field could move to a more standardized animal model, maybe one closer. I know it's a big ask actually, and I don't know the answer to how to do it, but canines and mini pig are better matches to human physiology. We actually see it in the parameter space. You can actually, if you look at all the parameters of the model, I mean, it's not shocking that rodents are far removed from where those other three are. So I'm not giving you anything insightful by saying that the pig and the canine are closer to humans. But if there's a way of standardizing the field, it would really increase accuracy. It would accelerate innovation further. It's something worth discussing.
Okay. Perhaps one question to Michael Weiss also, what would you need to move on to clinical development?
I think that the thermalin analog that I mentioned in the very promising pig trials overseen by Dr. Michael, who's with us today, are very close to being ready for clinical trials, and it would only require the investment to do so.
Right. And I wonder, during your talk, I mean, obviously, ultra stable insulins are really needed, in particular in countries where refrigeration is a big issue. But I also wondered if they could help for novel insulins. For instance, I mean, oral insulins, it's still a challenge to develop insulins that can be absorbed quickly and that are stable in challenging conditions like low pH.
Yes, so conformational fluctuations in the monomer enhance rates of proteolysis for a variety of proteases. We haven't tested them under the stomach conditions, but that's a general principle of the physical chemistry of insulin. These conformational fluctuations can be enhanced by substitutions like LIS-Pro, and so having ways to mitigate the dynamic instability of insulin could facilitate oral insulin development. Thank you.
And I actually, when I watched the presentation of Professor Chou on the mini insulins, so I mean, usually if something is small, it can be absorbed quicker. So do you think you could basically improve the onset of action or the onset of absorption, the speed of absorption with your approach?
Yes, exactly. That's exactly what we are trying to demonstrate these days. Going back to the same reason, I think there's one of the speakers or the moderators talking about the case of LIS-Pro where they still need to add zinc ions to promote the stability. That's actually the same challenge we are facing currently in terms of the monomeric insulin. Basically, by making it monomeric, things can move in faster. In fact, I think in early 90s, there are studies where they tried to basically label different insulin analogs with different association states and trying to see how fast they get into the bloodstream. And it seems like there's an inverse correlation between the speed of onset, PK-wise, versus the association states. So I think it's pretty clear that one can achieve that. But really, I think the challenge would be there's a reason why insulin was designed to be an hexamer, because that can really increase the stability. And then that's also what we are trying to work on. So I guess this will be related to the ultra stable insulin concepts that Professor Weiss discussed about. So what we are trying to do in this case, because we have already done modification, try to make it monomeric. So in our case, actually, we are using an excipient approaches. So we have some excipients that we believe that can stabilize these novel insulin in the mini insulin case, but also potentially for native insulin as well. And then some of you may be familiar with my colleague here at Stanford, Eric Apple. They are using some sort of polymeric based excipients for the same purpose. And then so I think sooner or later we will be able to address the stability need to basically to demonstrate a faster onset. And I think if I may argue, I believe the onset problem is more or less addressed by these excipients used either in the case of FIASP or the ultra rapid LIS-Pro, in a way that basically one can add a vasodilator, try to make things getting faster. So I think this is going to be working okay. I think the real challenge here would be something that can lead to a faster offset. And then I think the data showed by Dr. Weiss in their case where they can see a faster offset in the action of their ultra rapid insulin. I think this is probably the direction to go in the future.
Thank you. One question I had to Dr. Strano, because we initially in the first talks before the break, I think we discussed the glucose sensing part of glucose sensitive insulin quite extensively, and obviously it's very hard to really find a good glucose sensor, and I wasn't sure how this is integrated in your model. So do you just assume a good glucose sensor, or because probably none of the glucose sensing insulins we will have, at least in the nearer future, we'll have a perfect glucose sensor.
By glucose sensor, you mean a receptor that binds to the saccharide like a boronic acid? Is that what you mean, or do you mean metrology of human glucose measurement?
No, I mean the former. So basically something that senses the glucose level and then releases insulin or recaptures insulin.
That's a good point. And if are there the one way to ask your question is that are there chemical restrictions to our ability to bind molecules like glucose built into the chemistry? We have not mapped those on, but it's a great question. Nature builds carbohydrates when it does not want things to bind. So carbohydrates, they coat bacteria. It's an interface that these are molecules that nature does not use for binding. And in fact, if you search for if you do directed evolution or you look for molecules that can grab onto saccharides, it's hard to find. It's hard to find really low dissociation constants, and natural systems tend to just look for small fluctuations in carbohydrate binding. That's how a lot of glycoprotein signaling works. So it's a good question, and I don't know that I'm answering it well, but I think what I'm getting from your question is we could further refine these parameter spaces by putting in chemical constraints. How well can you dream up your ability to latch onto glucose under physiological conditions? And so I'll take that back to the team. We will think a lot about that. Yeah.
Great, thank you. And perhaps I can forward that to Dr. Weber. I mean, the question to me really is, you talked about the challenges of finding something which is specific to glucose and reacts fast enough. How much is toxicity an issue? Because I know that the PVAs also might have some toxicity issues, and I guess that this idea of microneedles or encapsulation also, they of course would solve the toxicity issues, but they might add to the time delays that then pose further challenges.
Yeah, I think the toxicity of phenylboronic acids and related chemistries are still to be determined. Probably there's some knowledge existing at Novo and other sorts of places on kind of the history of this space, and I know they've put them on evidence, and from almost 20 years ago in patent literature, at least putting these on insulins and different things. And so there may be more knowledge than I'm aware of, but I would definitely still have some remaining concerns on the toxicity of the conjugates, especially for something that goes in the body, right? And so if it's on the insulin or on a protein excipient, a polymer excipient, or some other sort of thing, I think the contrasting that with the benefits of, say, Professor Gu's microneedle system, at least you can pull it out of the body. And if you can actually have a fully polymerized microneedle with no free monomer, no other sorts of things in there that can cause toxicity as it's kind of leaching out, I think you could potentially avoid systemic exposure to some of these compounds that might be a little bit nasty. I think so. Removable devices would be useful in that regard. But like you said, then you have the issues of maybe the pharmacokinetics are kind of unfortunately delayed when you start putting them into these other devices, as the device has to swell and you have to get through sort of the transcutaneous region and whatnot. And so I think that there's certainly going to be concerns there as well.
Yeah, perhaps I forward that also to Dr. Gu. Yes. Because I mean, I'm coming from the continuous glucose monitoring. We were also discussing the delay between blood glucose and subcutaneous glucose, and in a way, the microneedle approach, I guess, will have the same issue, right?
Yes, actually, it's a very nice question, and we do actually identify the delay associated with the microneedle array patch, and as Dr. Becker actually mentioned, that the interstitial fluid associated with the dog, associated with mice, associated with pig, actually different, and also for human. So that's why we really want to further test on the human to identify if such delay can affect the overall performance of the microneedles. But based on the ongoing specific projects associated with the mini pigs, associated with the rats, actually we somehow didn't identify the significant delay of the insulin effect. Yeah.
Right. Now we also had a question to Dr. Weber. So what do you make of differences in your ITC binding isotherm data of the DPPA between glucose having 6 to 8 millimolar equivalents to saturation versus GDL diol saturating at 1.5 equivalents?
Yeah, I think this is something that Professor Chou alluded to and is also alluded to in the chem draw we have. I suppose it has something to do with the DiPBA having a strong preference for binding to the furanose form of the glucose ring, which is not really the majority species. And so I think probably the saturation is a little bit to do with that furanose equilibrium state between the two possible glucose forms. And so that is a little bit of a consideration, right? Is that, if it's only able to bind to what might amount to 10% or less of the overall distribution of glucose structures, you're not actually able to sense a lot of the glucose that's there just by virtue of not being able to bind it. So this is a little bit of I think an ongoing debate in the field, but I do think that IPBAs are primarily binding the furanose, and so that's going to limit the amount of glucose they can actually reasonably bind.
Okay, thank you.
By the way, so I just want to add one more associated with the toxicity of the PBA. One is just like the patch, you can get the materials outside the body, and this is one approach. And nothing is recently we actually started a very systemic study associated with the long-term toxicity of the PBA, injected, subcu inject, also oral delivery associated with the PBA-based materials. But so far we haven't found let's say the significant toxicity also. It's actually our students actually quite excited about that. Originally we indeed just feel uncomfortable regarding the toxicity, but this moment quite confident regarding the PBA's biocompatibility. Yeah, but long-term evaluation is still ongoing. We really want to for example, subcu injection every day for over half a year, this kind of test. Yeah.
Right, thank you. I wanted to move to one other topic, which is quite important to me because we saw different approaches, of course, of glucose-sensitive insulin. So the initial idea was basically, well, insulin is released in case of high glucose and then is recaptured in case of low glucose. Of course, with some approaches like the microneedles, that won't work anymore. So the insulin will be released, but you can't get it back in any way. So I wondered, although it's not directly related to your talk, Michael, Michael Weiss, how important is it to get the insulin, so to have some hypoglycemia protection?
How important is it? Well, I think that it would enable the patient or the algorithm to be more aggressive in treating hyperglycemic fluctuations if we didn't have to worry about hypoglycemia. And in really elegant recent simulations from Michael Strano's group, we see this, even modest degrees of glucose responsiveness confer protection from hypoglycemia at increased dosage that enhance time and range. So I think the ideal glucose responsive insulin that's ultra rapid would be of great value in fully automated closed loop systems.
Perhaps on the same topic to Dr. Mahdavi, I mean, you presented also the glucose-sensitive glucagon. That of course would be able to, with some kind of biohormonal approach, to prevent hypoglycemia, potentially more reliable than just releasing insulin. So is this why you're looking into that, or is it just different, just for hypoglycemia prevention?
Yeah, it's a great question, Tim. I think it's both. So I think if you can prevent hypoglycemia, you can also be more deliberate, or I guess be more intense with your insulin injections, and have better glycemic control, overall glycemic control, and no chance of hypos. So, you know, I think, I'm not sure if that answers your question, but really the way we think about it is simplifying the overall strategy, right? How do we simplify insulin therapy? And within that context, we think glucose sensing glucagon is a very valuable tool, and one that doesn't get as much attention. So my thought on that is, you know, if the panel is interested in concepts in insulin therapy that are really transformative, that's where we should be thinking. Because I mean, I think to what others had mentioned, there is now a couple of programs that are very advanced in glucose sensing insulin, and really the question now becomes, what's the next thing that will be quite interesting? And I think the glucagons are going to be quite interesting. So, so in that context, I think glucagon is really interesting. I think in another way that's it's really interesting is the ultra-rapid insulins that you know, Danny just talked about. If you could have those in a simplified format, or Michael mentioned, you know, these insulins in simplified formats where you can inject them with fixed dose, then, you know, a glucagon that kind of counteracts if you overdose on that is ideal, because then you can have a really simplified strategy for your mealtime and just have a basal on board. So that's another way to kind of think about this idea. And of course, if you had glucose sensing insulin that will be in the clinic and hopefully approved soon, then a glucagon can really be a great, you know, complement to that.
Yeah, perhaps the same question to Dr. Gu, because you also work on some kind of biohormonal approach.
Yes, I think, you know, the glucagon based delivery system is also very clinically like meaningful. It's just like you know, the dual pump, like to deliver insulin and glucagon, and such, for example, glucagon patch can serve as a safeguard, you know, for the insulin delivery, especially can be put on like during the night time, and can, you know, enhance the safety of the insulin usage. Yeah.
Thank you.
But meanwhile, you know, I just want to add two things, like just simply a glucagon patch without any like a glucose responsive component still can be meaningful. I mean, just simply put on, okay, and it could be relatively easily translated. Yeah.
Yeah, great. There's one question from Simon Heller. Perhaps you would ask that, Simon.
Yeah, thank you, Tim. And actually, while I'm online, I just want to apologize to Dr. Michael Weiss for not introducing him properly. It's a very short notice because of technical difficulties, and it was ungracious of me, but I didn't have these details. I have now, and perhaps I just quickly let the audience know that Dr. Weiss has done fantastic work in this area. He now works at the Indiana University Precision Health Initiative and had a long and very prestigious career at Case Western Reserve, having worked at MIT. From his talk, he's extremely knowledgeable and a leader in his field, and I want to apologize to him personally for not acknowledging that, but I didn't have access to his CV at the time. But I'd like to ask about the in silico work, because it looks very exciting. And as somebody who's been using imperfect insulins for 30 plus years, the frustration is how long it takes, as we heard actually from Peter Kurtzhals this morning, to bring an insulin to market and give insulins to patients with the benefits we have seen, and indeed what we hope will be even better. So does in silico offer a route to much shorter regulatory approval and getting therapies to patients quicker? And I'd really be interested in Professor Strano's views on that, and indeed others, because it is really difficult for us waiting for years when we see the papers which offer great promise.
Was that directed to this Michael or the other?
Michael Strano, I said.
Yeah, thank you. It's a tough question, and I don't know that I am I'm very humbled actually to be in this space. Maybe I would want to defer to my colleagues on the timeline of development, right? I think it's related. Well, can I ask more about the? Let me defer to my colleagues, because I think it's the timeline of development. There are people on this call that have far more experience and are more knowledgeable of how that works. We think we have part of it, really a tool to put in your hands that can help. Is this a?
Yeah, well, maybe actually we've got members of the scientific committee, but again, people like Michael Weiss and others who've been working on this.
Yeah.
It's the work is tremendous, and it offers promise. But we are now going to wait another 10 years till we see these products developed, or can we see ways of shortening what is a very robust but lengthy process?
Yeah, and I think it's also fair to say that we are getting more and more into looking into in silico data, and as so are the regulators. So I mean, there are now lots of models that FDA use, and I think that will hopefully help to get to approval faster. But I guess it's also very important to really pick the right candidates quicker or more or better, because as we heard from Peter Kurtzhals, I mean, you have so many candidates that won't make it. So it's very, very difficult to really pick the most promising candidate, and so we hope that simulation will help. So there's actually one quite critical question from the audience, which I like. So the question is, does the panel have a view on why insulin glulisine, so Apidra, which is a zinc-free monomeric insulin, but it only has a PK profile that is not faster than that of hexameric rapid-acting analogs, or in fact, it appears to be slightly slower. So the question is it possible that focusing on monomeric insulin is not the right way to go, and other promising approaches shown in ultra-fast products in human clinical development should be the main focus? So I'm not sure who would to handle that first.
I can try to address this question a little bit.
Great. Yes, please.
So I think this is a terrific question in terms of trying to understand whether one should really use a monomeric insulin as part of the product profile to achieve something that's ultra fast, both onset and offset. So I think it's correct to say that, I mean, in the Apidra formulation, there's no zinc there, and potentially one will argue that that's something really going to give you a more monomeric form. But I think we should kind of take one step back. So lots of times when we are talking about these association states, we need to talk first about the monomer and dimer association. So what I would say, let's use the case of this pro, can really do is, because of flipping the two residues, they actually can decrease the dimer association state quite a bit. And that's why, potentially in even higher concentrations, it could be even more monomeric. And of course, because of stability reasons, they need to add the zinc there. So what's in the vial is still hexameric. And again, in the Apidra case, yes, I don't think they add the zinc, but basically for native human insulin, for a lot of these insulin, if they still have the dimer interface, in high enough concentrations, they still form hexamers even without zinc in the vial, because if you have enough insulin, they will still basically go to a form that's more stable. So I will say that basically we will need to, because it's still a very high concentration in the vial we have. So for U100, this will be more than 3 milligram per ml concentration of insulin out there, which is basically way over the dimer association state. So I will argue that we probably still need to talk about the specific concentration they are using, and then the dimer association, the dimer association constant in a way that to provide a kind of fair point of view for the comparison. So kind of going back to the original question, and then I will refer to the kind of the study I cited a while ago in a way that I think there's a pretty clear evidence if one can really achieve both stable and monomeric insulin, this is probably the direction to go, but that's probably still the challenge to address. And then going back to this specific case, it's basically a simple kind of mass transfer question in the way that larger genes were getting faster. Larger genes were getting slower, small genes were getting faster. And one of the key comparison we can probably talk about, and some of the panelists referring to the artificial pancreas workshop last week. So some people talk about polymatide. So that's an amylin analog. Basically, in terms of the size, it's actually not something too big. Not much bigger than insulin, and it's still going for subcutaneous injection. But it has a much faster PK and then faster onset and the faster offset. So I think that will be a good molecule for us to remember that, because it is purely monomeric, and that's something even in a subcutaneous injection one can try to achieve. So I will say that potentially this is something we could think about.
Great, thank you.
So yeah, I'd like to agree with that. Lispro and Aspart do not have proline at B28, which is key to the dimer interface. Blue lysine has proline at B28, and fortuitously, the change in charge at B29 confers resistance to fibrillation in the absence of zinc, which enables a successful formulation. But the dimer interface is intact, and the trimer interface is intact. And my speculation is that at the ionic strength and complex environment of the sub-Q space, that it is self-associating, and it's not a monomeric insulin analog functionally.
Thank you. There's one final question to Dr. Mahdavi, because you mentioned one topic that we neglected a bit, perhaps during the symposium, of having an insulin that primarily acts on the liver. So I think in your development path for glucose-sensitive insulins, you indicated that at some later point, we might have a hepatopreferential glucose-sensitive insulin. And that was something that I was very much interested in, because we solve a lot of issues with glucose-sensitive insulins, but it's still the issue of giving it in the wrong place. It will primarily act on muscle and adipose tissue. So how important is that, and how difficult is it to achieve? There are not a lot of promising developments for hepatopreferential insulins right now.
Yeah,
I would like to beg the pardon of my colleagues. I have to jump off to another meeting. I wanted to thank the panelists. Thank you very, very, very much.
Thank you, Michael. Thank you so much.
Thank you.
Thanks, Mike. Yeah, Tim, it's a good question. Let me answer it in two ways. And I think it relates to the discussion we just had on ultra-rapid insulins, because I think if you, first of all, I think any insulin that's developed, it's important to consider the market that it will address for it to be a commercially viable product. And I think if it's just hepatic preferential, probably is just not enough there to advance a molecule like that, if that's the only aspect. I agree with Danny that in the ultra-rapid space, you need more than just ultra-rapid. It's got to be ultra-rapid and stable and maybe have some other characteristics. But I think if you have a really good glucose sensing insulin, you will get both of these effects covered in a sense. And the reason I say that is a really good glucose sensing insulin that's available in circulation, if it responds quickly enough to glucose fluctuations, then as glucose levels go up, it will respond faster than any ultra-rapid insulin can ever respond to glucose spikes. And the other aspect of it is that we know that glucose levels initially go up in the hepatic portal vein. So if the speed of activation is fast enough, you actually could get a preferential hepatic effect purely from the mechanism of activation of that glucose sensing insulin. And so I think that's where what I mentioned about the reversibility of these systems and making sure they have fast reversibility is important, because I think in systems where you have the need for diffusion from a depot, or like as you mentioned, from a patch, then obviously that's a very different proposition. And I'm not sure if the activation speed in systemic circulation is there exactly the same way. And I'm not sure if you could get the preferential hepatic effect unless the insulin that's in there is already preferential hepatic to begin with. Then it's difficult to envision that speed being there. But I think in systems that are already in circulation and fast enough, then I think you can get coverage of what would be required from an ultra rapid in terms of speed.
Right.
Nevertheless,
go ahead.
Nevertheless, I think there is like a good case to be made for an ultra rapid that has many other characteristics like stability, etc., as Danny mentioned. I think it's just how these things play in time, right? When would we have an insulin that has that speed and come on the market versus other products? So I think the challenge is the timing of these things with respect to each other.
Yeah. Thanks very much. Unfortunately, we have to stop here. I could go on for hours, but I really want now to pass on to Rachel Connor, who is the Director of Research Partnership at JDRF UK, for concluding remarks. But of course, I also would like to thank all the speakers for great presentations, a very good discussion. And I learned a lot about challenges, but also about potential solutions. And I think Rachel will tell you a bit more of how we are going to proceed. Thank you so much.
And could I just echo, Tim, your remarks that I also, as one of the co-chairs, want to thank, because outstanding presentations, really great discussion. And Rachel, I hope this will be a springboard for doing some really good science in this area.
Thanks so much, everybody. I will be brief because I know we're at the end of a long agenda, but I just wanted to lay out what the next steps for the Grand Challenge Partnership will be. First up is we're going to make videos of all of today's presentations available online, so that those colleagues of yours who weren't able to join us today will be able to catch up on the fantastic presentations and insights that we've received today. We really want to spread the word as wide as possible about the fantastic work that needs to be done and is happening in the novel insulins field. As you heard from Simon earlier on today, we are hoping to distill all of the knowledge and insight that was gained from today's symposium into a review paper, which we will hope to get published, and again bring more scientists into these conversations. But it's really clear that we need transformative new insulins based on bold scientific ideas, because people with type 1 diabetes need lots of things from insulins that the ones we have today just can't deliver, whether it's glucose responsiveness, liver targeting, ultra rapidity, thermostability. All of these things are needed to make life for people with type 1 diabetes much easier. It's really been wonderful today to see engagement from speakers who are based both in academia and in industry coming together. I think all of the speakers today have shown such real passion and drive for developing new solutions for people with type 1 diabetes. And by bringing those strengths together, I hope we will really be able to meet that goal that we've all joined together in setting that bit faster. I can't say it often enough, really, we do need new insulins. Some people with type 1 diabetes are doing a phenomenal job in managing glucose levels exquisitely, using the best available technology and information, but it is a job, and it is a job that no one asked for. Making insulins that work better isn't a nice to have. It's absolutely vital if we're going to improve quality of life for everyone who lives with type 1 diabetes, but especially so for young people with type 1 diabetes, who, as the data tells us, have the toughest time in managing their glucose levels. So with all of that in mind, we will be moving to launch an opportunity for all of you to apply and pitch for Grand Challenge Partnership funding to push forward in developing those new products. Thanks to the generous donation from the Steve Morgan Foundation, we have a budget of £15 million, that's UK pounds, to do this. And we are looking for transformative ideas. This is not the funding call for incremental steps. This is about bold new visions. Based on what we've heard today, the scientific advisory panel members who you've met today and those who weren't able to be with us will be reconvening, shaping this funding opportunity a little bit more, but we will be coming out with the mechanics of this opportunity as soon as we can. If you signed up to allow us to contact you when you registered for today's symposium, you will be the first to know about the opportunity. It will hit your inbox. If you didn't sign up to give us those permission, please keep an eye on the Grand Challenge website, which my colleagues will be putting a link to in the chat as we speak. And it's coming up on screen, there we go. And you can also keep an eye on our Twitter and our LinkedIn feeds as well to make sure that you don't miss out. So I'm going to close just about on time with some quick hurried thank yous. But thank you to all our scientific advisory panel members who've so generously given their time and advice to the Grand Challenge Partnership so far. All of the speakers who were just phenomenal. Thank you so much. I'd also like to thank my colleagues at JDRF International, Dr. Sanjoy Dutta and Dr. John Rosen, for all of their help and advice as we've been moving through this. And of course, my colleagues at the Steve Morgan Foundation and Diabetes UK, as we've moved through the partnership. But the final thank you must go to my colleague Eleanor Kennedy, who you haven't seen on camera today, but she is the one who has pulled together this fantastic agenda for us and made sure that she has worked across multiple time zones to get everything together for today. So thank you to Eleanor. And with that, I can close the meeting, allowing you all to find a beverage appropriate to your time zone, whether it's your early morning coffee, your evening glass of wine, or your bedtime cup of tea. And with that, thank you all so much.